right, welcome to Standing For Truth. My name is Donny Budinsky. I am your host and moderator for tonight's intellectual showdown. It is a privilege to guide this epic clash between Dr. Zach Hancock and Dr. Jerry Bergman. Tonight, the truth goes to war. One stage will become the battlefield of ideas. This is not just a debate, it's a battle of the minds. Now, the question for tonight is one that shakes the pillars of history. Can evolution explain the arrival of the fittest? The countdown has ended, the stage is set, and I'm sure it's set for a debate to remember. Gentlemen, I am pumped and thrilled to have the both of you here for this exchange. Let's break the ice a little bit, get acquainted, as I said. Dr. Zach Hancock, let's start with you, as it's not your first time here on the channel, but it is your first, if I remember correctly, formal debate here. And so I really appreciate you offering your time for this. And let's hand it to you for a little bit about yourself, if we could, Dr. Hancock.
Yeah, thanks so much, Donny. You make an excellent hype man. I feel like a gladiator entering an arena right now with that introduction. So I'm Zach Hancock. I'm an evolutionary biologist. I specialize mostly in population genetics, phylogenetics, genome evolution, that sort of thing. I do a little bit of theoretical work, a little bit of empirical work. I like to kind of think of myself as a jack of all trades. I am currently an assistant professor at Augusta University in the Department of Biological Sciences. Before coming here, I was a postdoc at the University of Michigan for 4 years, mostly doing population genetics. And then before that, I got my PhD at Texas A&M University in ecology and evolutionary biology, mostly working in crustaceans. So looking at their genomes, how they evolve. And then I did a little bit of theoretical work there as well. I also run a YouTube channel where I do some evolution education. It's called @TalkPopGen. Evolution education, a little bit of talking about pseudoscience, which we'll talk a little bit about today, and then as well as some history and philosophy of science. So I'm really pumped for this, really excited for it. I'm very happy to be here.
Okay, very good. I appreciate the energy, Dr. Zach Hancock. Thank you for the intro. For those that want to see more from you and what you're putting out, they can check the description box of this video. I've got a link to your channel. And so, gentlemen, I know you're more than equipped and qualified, clearly, to be discussing tonight's topic, and I'm looking forward to it. Dr. Jerry Bergman, it's great to have you back as well. Not your first time here on the channel, nor is it your first formal debate here. Just a few months ago, time flies by when you're having fun. You were here for an excellent debate with Gutsy Gibbon on human evolution. So I appreciate you giving your time yet again for an important debate. Jerry, tell us, how have you been since the last time you were here, but also tell us a little bit about yourself.
I've been busy, and I keep too busy, my wife does, but that's, I guess, the way it is. I tell her it's better than spending my time in bars or other places. So at any rate, I've been busy, and I have a feeling this will be not so much a debate, but a discussion. Later on, I have a few questions to ask, which I think a population geneticist can help me answer. So I'm looking forward to that. But I used to be an evolutionist and an atheist. In fact, I worked fairly well with, very well, actually, with most atheists. Marilyn Mary O'Hare was my boss, and she published everything I wrote, and she had often good things to say about me. She ended up with a tragic end in her life, but I guess that's part of the risk you have in some of the people she associated with. But at any rate, I'm looking forward to it tonight. And I've done a number of debates in many venues, so here we are.
Here we are, Jerry. A lot of people have been excited for this. I've been excited for this. And I have a feeling this will be a debate or discussion to remember. And so for those that want to see more from Jerry Bergman, please check the description box of this video. And I've got the relevant links where you can find more from him. Okay, gentlemen, I appreciate those introductions. And again, I appreciate the time that you gentlemen have offered, not only for the debate, but the time leading up to the debate. I know these require a lot of prep and study. And so thank you, gentlemen. Let's go over the format. And so we do have a comprehensive event for tonight. Again, the question is, can evolution explain the arrival of the fittest? Dr. Zach Hancock takes the affirmative. Dr. Jerry Bergman takes the negative. We have 18-minute opening statements, starting with Zach. Then we have rebuttals, so plenty of time to interact and engage with each other's opening statements, directly answering questions and arguments brought up in the opening statements. 10 minutes each for that. Then we'll jump into a discussion. The discussion will be 45 minutes. Rather than a real strict cross-exam, this will be more free-flowing and organic. But I do also want to make sure that our guests tonight, our debaters, Zach and Jerry, have more than enough time each to pick topics and ask the questions that they'd like to ask. Then we have closing statements, 5 minutes each, good opportunity to wrap up our thoughts, wrap up our points, and address anything we feel has been left hanging up to that point. And then this is where we get you guys in the audience involved. We're going to have an audience Q&A. We're flexible, probably somewhere between 25 and 40 minutes, depending on how many questions come in. And so please, if you have a question on tonight's topic, make sure you're tagging me at Standing For Truth or at Donny. That way I don't miss it. And let me know who the question's for. Okay, Dr. Zach Hancock, we are going to get into our first opening statement. Again, the opening statements are 18 minutes. And so whenever you're ready, feel free to get your presentation up if you do have slides. And I do see that at the bottom right now.
Let me know when you see it.
And gentlemen, I can also, to help you out, I know how quickly these go by, I can give a 1-minute warning when we reach the 17-minute mark. And that way you'll know to start winding things down. Okay, Dr. Hancock, it is up on the screen. Can evolution explain the arrival of the fittest? The floor is yours. Feel free to begin whenever you're ready.
Okay. Thank you so much, everyone, for being here. As I said, I'm very excited for this. Thank you so much to Donny for suggesting this, and thank you to Dr. Bergman for agreeing to do it. I think this is a really important topic. It's a really interesting topic, and I'm very excited for it. And so the question is, can evolution explain the arrival of the fittest, right? And of course, I take the affirmative, but you might be curious to know where this cutesy little term comes from, the arrival of the fittest. So it's often accredited to Hugo de Vries. It goes back a little bit further than this, but he famously said in 1904 that natural selection may explain the survival of the fittest, but it cannot explain the arrival of the fittest, right? So what does he mean by this? So he means, in effect, that natural selection sort of sorts through the variation within populations. It keeps the good stuff, it purges the bad stuff. It's kind of a sieve, right? But natural selection doesn't cause new things to occur, right? So where does the new variation within populations actually come from? Now, those of you that are familiar with history know that Hugo de Vries actually had the answer to this question, and it's going to be the main topic that we'll be talking about today. But I think it's important for us to sort of situate ourselves within evolutionary theory. What is it really? Evolutionary theory is fundamentally about what happens to variation in population. So no two of us are alike. We're all different. But where does that variation come from, the origination of it? And then what happens to that variation after it emerges? That's the focus of evolutionary theory. So where does variation come from? It comes from development, so little quirks that happen during development. It comes from the environment, different things that you're exposed to. But those two things often aren't passed on to your offspring, right? They're not heritable. The heritable things within evolution are things like gene flow. So you're getting new genes from other populations being introduced, adding to the variation. Or if you're really interested in where brand new variation comes from, never before seen, that's mutation, right? And that was Hugo de Vries's mechanism, was mutation. Now, mutations are then filtered into the next generation by two other processes, and that's genetic drift and natural selection. And we call these sorting processes. So genetic drift just kind of sorts that variation randomly, and natural selection is trying to favor variation that increases reproductive success, and it's purging the variation that reduces reproductive success. And this process leads to different variation at the end. And this is a constant sort of feedback mechanism where variation is produced, it's weeded out, and then it's being reintroduced again, right? And so the key that we're going to focus on here today, all of the structure of evolutionary theory, what we really care about are mutations, right? This is the ultimate source of novelty in evolution. But mutations are one of the most misunderstood concepts in evolution. And if I say, what are the effects of mutations, you can get two quite distinct ways of answering that question. So I want to break that down. So one way of answering it are by looking at what are called fitness effects. So fitness effects are about the rate and the probability that that new mutation spreads through the population. In effect, it's a measure of how does that mutation impact the reproductive success of the population. Now notice that this is a fundamentally different question than what is the biochemical effect. So does this mutation generate a new protein? Does it generate a new protein interaction, right? Like what is it actually doing biochemically? And note that you can generate a new protein and reduce your reproductive success. You can break a protein and improve your reproductive success. So these two things are not equivalent, and we need to be sure that we're keeping them separately as we're talking about mutational effects. Furthermore, it's important to remember that biochemical effects are intrinsic. They always sort of are doing the same thing, but fitness effects are extrinsic. They're determined by the external environment itself. So if in one environment, perhaps you have the most optimal phenotype, so you're up here in the A bullseye, sitting right there in the middle, any mutation is going to move you away from that optimum. However, if you enter a new environment or the environment changes, those exact same mutations might pull you closer to the optimum. So it's important to remember that the effects of mutations are not intrinsic, but are extrinsically determined. However, I don't actually think that this is what we're interested in. When we're talking about mutations, what we really care about is how do we get new stuff, right? How do we get new biochemical reactions and interactions, right? That is, how do new genes evolve? And fundamentally, I think what creationists really care about is how do these new things actually generate new body plans, right? So how do we get a C. elegans, a Drosophila, a Cephalochoridae, humans, etc. And so this map kind of lays out the developmental network of genes, regulatory interactions, and protein-protein interactions of how all of these different critters are constructed. So the task before us is to show that mutational processes can generate new proteins, can generate new protein-protein interactions, and can generate new regulatory networks, right? So all of these different components can be generated by mutation. And then lastly, we'll reintroduce that fitness component to tie it all together. So starting off, case one, I want to show you the Darwinian mechanism. So that's what I'm calling it, is the gradual step-by-step mechanism. So what we see here are two different sets of proteins. So this is protein E2, protein IM2, and then E9 and IM9. And these sets of proteins have to bind together, they interact with each other, and they form an endonuclease. And this endonuclease serves the functions of immune function in bacteria. Now, these two different sets of proteins differ by 17 amino acid substitutions. They have different backbone structures, they have different hydrostatic interactions, the configurations are different, they're very different proteins. And yet the Darwinian mechanisms suggest that they are, since they share a common ancestor, we should be able to walk one mutation at a time back to that common ancestor, and then up from that common ancestor to the other protein pair, all without disrupting the function of these proteins and maintaining the binding of these proteins. So this is a kind of tight walk that we have to pull off here, right? So if these two proteins really share an ancestor, one by one, these mutations should always lead to functional proteins. Is that possible? Can we actually do that? Well, it turns out that's exactly what these researchers did. So they took these original two proteins, they mapped out every possible mutation that could happen to them, and then they said, are any combination of those mutations, can we bridge these two different variants? So these are the two proteins here. We start off right here along the x-axis is changes in the E2 protein. The y-axis changes amino acids in the IM2 protein. All the red squares represents viable cells, so cells that are still able to build that endonuclease. And sure enough, all these arrows represent step-by-step amino acid changes. And then when you step over these green lines, those represent swapping your interacting pair. So you can not only change that one protein, but as you move across them, you're now binding to the different one. And so all along this path, you never lose function. So new functional proteins and new protein-protein interactions can be maintained and evolve in a step-by-step fashion without ever generating a functionless protein. The second case I want to talk about are gene duplications. This is the Onoian way of getting new proteins. So you start off with gene A, and this gene has these three functions. These are these black squares. That gene duplicates, and now you have a daughter copy, and that daughter copy is just redundant, right? It's doing the same thing that the parent copy did. But over time, since it's redundant, mutations can occur. It can acquire a new function, now this other block, that's different from the parent, and we call that neo-functionalization. So I want to give you two big examples of this. Now, this first example relies on us all agreeing that these two plants share a common ancestor. Now, I suggest that, all right, suppose many of you probably don't know what these two plants are. So these are wild sorghum and wild corn. Yeah, this is what corn looks like in the wild. They are one another's closest living relative, and they are very, very similar genetically. So I assume we can agree that they share a single common ancestor. Now, the interesting thing about these is that since they diverge from each other, corn has undergone a whole genome duplication. And so if you take the sorghum genome, you take corn's genome, and you can align them, there's a third genome that's much younger that also aligns. So there's an extra genome in corn. And what this means is there's a lot of extra DNA in corn and a lot of DNA for mutations to play with and to build new things with. So these researchers looked at just the regulatory networks. They were curious, is there new regulatory pathways, new regulatory genes that have evolved since this duplication? And sure enough, they found 13% of the duplicated regulatory genes had acquired new functions. So let's put this into numbers. There are 32,000 protein coding genes in sorghum. If you need, on average, one regulatory gene per protein coding gene, that means that since corn and sorghum diverge from each other, corn has evolved 4,160 brand new regulatory genes. If you think they share a common ancestor and split since the flood, that's more than one new gene per year since they split from one another. Here's a similar sort of example. So these are two fruit flies. They are of the same genus, right? They're both Drosophila. And what these researchers did is they also looked at duplicate pairs. So they looked at genes, duplicates that melanogaster had that Pseudoobscura didn't, and vice versa. And then they wanted to know, did these daughters take on functions not present in the parental copy? They found 281 duplicate genes that had new functions that were different from the parent copies in either of the different species. And the way that they determined these new functions is they looked at differences in protein-protein interactions, as well as differences in the expression levels and what tissues those proteins were being expressed in. So 281 novel daughter copy genes that had evolved since these two flies split from each other. Now, what's really the clincher to this is that older duplicates were more likely to show new functions than younger ones. Now, this is exactly what we would expect if the mutational process takes time and has to tinker with this daughter copy. So the young ones almost always showed the same function as the parent. The old ones showed new functions. And indeed, what they found is that the oldest ones also increased the amount of integration. So they were being expressed in more tissue types than the younger ones were, which tend to be kind of restricted in where they were getting expressed at. So now we've seen thousands of brand new, both regulatory and protein coding genes can emerge in relatively recent times within what is creationists would generally expect are the same sort of kinds, right? So now let's see if we can reintegrate this all together. Let's bring these different things together and see if we can reincorporate fitness with a sort of biochemical effect. And ask this, I think, really important question is, how easy is it to do this? Right? Like, how easy is it to get this brand new protein that actually results in high fitness? It's one thing to show that it's there. It's another thing to actually see the mutational path that can generate it, right? So what this study did is it tried to answer this question by generating the complete fitness landscape of one protein. So what it did is it looked at this one gene in E. coli. And this gene is essential. E. coli have to have this gene to function. So in a lot of cases, antibiotic resistance might emerge by breaking something. But this particular gene cannot be broken. E. coli must have it. So the way that to get antibiotic resistance for this particular kind of antibiotic meant that this gene had to not only maintain its original function, but it had to acquire an additional function on top of that was able to resist the antibiotic. So you start off with this protein, and then they used CRISPR-Cas9 gene editing, where they basically just created a gazillion mutants, right? 260,000 different novel mutational combinations of this protein. And then they subjected these bacteria to a mass selection experiment, right? So they just blasted them with antibiotics to see which one of the mutants were able to survive these different treatments. They then took those mutants and they deep sequenced them. They sequenced all of their DNA, looked at all of the variation that they had, and then they created fitness landscapes. And a fitness landscape is, in effect, you can think about it like a topographic map. And so every single point in that map is a different mutational combination, right? So it's a different trait or a different genotype. And areas of that map that are very low, that are valleys, or what we call areas of low fitness are valleys, and then areas of high fitness are peaks. So certain combinations can generate high peaks or low valleys, vice versa. So how many peaks did they find in this landscape? And this tells us something about how easy it is to evolve this protein. They found 514 fitness peaks. So as you went out across this fitness landscape, 514 little hills where you could get a better protein combination. Most of them were relatively low fitness, but 74 of them had a really high fitness. So you just needed to get to one of those 74 high fitness peaks to be able to resist the antibiotic. Now, importantly, that tells us something really cool, and that is that there's a lot of ways to acquire this new function. Right? And this is not unique to this study. There's lots of studies that found that proteins are actually able to do lots of different things and get to this in a fairly easy fashion. So how easy is it to get to this? So all 74 high peaks of fitness had what's called a really large basin of attraction. And what that's a fancy way of saying that there's lots of mutations that will get you to one of these high peaks. And in fact, over 100,000 possible mutational paths would put you onto one of these high peaks of fitness. So it's not hard at all to find these fitness peaks. Unlike what is often claimed, you can actually arrive at these peaks really easily. And the way to kind of think about protein evolution is that there's many roads to Rome. There's a lot of ways to find functional proteins. So, can evolution explain the arrival of the fittest? One of the things that I led with is that creationists are often interested in these big body plans, like how do big new things emerge in organisms? And you might feel like I've only shown you little things, little examples of things. But what's important to bear in mind is that to get new body plans, effectively, you just need new genes, right? You begin as a single cell. Your heart doesn't evolve. The genes that build your heart evolve. So if I can show that new novel proteins can evolve, if new protein interactions can evolve, and if new regulatory pathways can evolve, then we have shown all of the pieces that are necessary to generate these complex body plans can evolve via the mutational pathway. Now, if we're interested in going into deeper examples of this and looking at sort of the evolution of these Hox genes and various things like that, very happy to talk about that further. But I think for now, what I've demonstrated is that each one of these features are capable of evolving via mutations in all the examples that I have provided. And what this means is that mutation is capable of explaining the arrival of the fittest. You can get new genes, you can get new proteins, you can get new regulatory pathways, and not just a few, but thousands of them relatively quickly. So thank you so much for your attention. I look forward to hearing Dr. Bergman's argument, and I look forward to the rest of this discussion. I think I'm a little quick, but I will cede the rest of my time.
Dr. Zach Hancock, thank you very much for that roughly 16 to 18 minute opening statement on the question, can evolution explain the arrival of the fittest? Dr. Jerry Bergman, I will hand it to you. But before I do, I want to remind the audience, anybody just joining us, we will be having an audience Q&A once we're done with our main sessions here. And so please, if you do have a question or a challenge or perhaps an objection to anything our guests are saying here tonight, do your best to tag me at Donny or at Standing For Truth and let me know who your question's for. And we will have some fun interacting with your comments in the Q&A period. Dr. Jerry Bergman, we are now going to reset the timer, and you also have up to 18 minutes for an opening statement. Feel free to get your presentation ready. I do have it up on the screen now. And Jerry, The floor is yours. Go ahead.
Okay, thank you very much. Zach did a good job, by the way. Reminds me of some of my courses I took in genetics a while ago. He's probably more up to date than I was, but nonetheless. Okay, Zachary and Daniel looked at Stanford's paper, wondering if there's a motivation. Well, there probably was. On the other hand, though, my experience is that evolutionists also have a motivation in doing their research. What's evolution? Well, here we go. Common picture from the apes to a modern man. And beneficial mutations, I've looked at a number of them, and it's kind of a summary, but Jack probably knows of other examples. But basically, they affect the enzyme's active site, number one. Number two, they often lose specificity so that they can break down other sugars than they were originally designed to do. And sometimes they break a gene, and that allows another gene to function. So these are some of the common factors that I've noticed that beneficial mutations arrive at. Darwin was concerned about evolution, obviously, and he says the species have descended from other species by insensibly fine gradations. Why do we not everywhere see innumerable transitional forms? Innumerable transitional forms must have existed. Therefore, why don't we find them? Why is not every geological formation and every stratum full of such intermediate links? Geology assuredly does not reveal that. Now, of course, people today would say, well, we found many, and I spent quite a bit of time looking at the fossil record, and I found that that is still a major problem in evolution. We simply don't have many. In fact, in many areas like insect evolution, we have no evidence of transitional forms or very little evidence. One of the most famous examples was called the progression from monkeys to man, and that has been touted out as evidence for evolution for decades, human evolution. And now more research has been done, and Dawson did an excellent book, and he shows that by and large, these are made up of fakes. A vast majority of them are not valid, and we basically don't have a progression. Now, he still accepts evolution, but he doesn't explain how we get molecules or monkeys specifically to man. So he still believes in that, but he says that the progression that we see has been falsified. And Alexander Wirth, another researcher, said the idea that evolution is a hierarchy of complexity with humans on top lurks in everything from biology classes to politics. He concludes it's time to unlearn this false and harmful view. And that idea, that awareness is becoming more and more common in the literature. Another comparison is between genes of chimps and humans. This claim here, 98% similar. More research has been done. A problem is, of course, comparing chimp genomes and humans is not easy. It's not simple. Nature, though, published an article which found 85.1% similarity for the autosomes they looked at. And I understand the sex chromosomes, there is even greater difference. But even 85% similarity is a huge difference. Specifically, about 450 million differences. How is this bridged? Of course, as Zach correctly brought out, by mutation. Ernst Mayr said, ultimately, all variation is due to mutation. Theodosius Dobzhansky, who's from Ukraine, by the way, in Russia, said mutation is the only source of the raw materials and hence of evolution. No mutation, this is an easy way of explaining this. No mutation, no evolution, period. Mutations are random, and beneficial mutations are rare, but some exist. I'm not denying that there are not any beneficial mutations. What I'm saying is that the vast majority are not beneficial. In fact, Stebbins said the frequency of useful mutations in a species has been estimated conservatively at 1 in 1,000 mutations. This is a well-known evolutionary biologist, and his conclusion is 1 in 1,000 mutations. And Mueller, the man who started most of this research in mutations, understanding mutations, Herman J. Mueller, got a Nobel Prize for this, by the way. The great majority of mutations, he said, well over 99% are harmful in some way, as to be expected from the effects of accidental occurrences. And the last quote, many others, but these are ones that I found were useful because they were said by a number of leading evolutionists. The ratio of expressed to harmful mutations is about 10 to 1. And the key is expressed, because many, of course, are not expressed. Now, my background is in medicine. In fact, I worked in cancer research. I had to get certified to work with radiation because, as was brought out in our courses very clearly, there is no safe level of mutations. There's no safe level of ionizing radiation. And we had to carry with us, we had to work with mutations and try to reduce affecting us. Ironically, though, in my department, doing cancer research, 2 of the professors died fairly young of cancer. Now, this may not be related to the work we did, but it may be. Obviously, cancer is one of the leading killers, and so that would not be surprising. What causes disease? Well, disease is caused by pathogens, diet, and toxins, and mutations. Somatic mutations cause cancer, and mutations of the genome, the gonad cells, which causes birth defects, again, are a result of mutations. Down syndrome, trisomy 21, of course, is one of the most famous examples. Dawkins tried to explain how evolution works. We start out with a bunch of letters, and the goal is, methinks, is like a weasel. So letters are mutated or changed, and then those who fit the goal selected, and after 44 changes, we get methinks it is like a weasel. Now, the problem with this is, of course, this is done by computers. I would show this to students, and they right away saw the fallacy of it, because we're not talking about mutations that you would expect to find in an environment and selected one at a time. In fact, selection could not occur in this case until we got the endpoint, which is methinks was like a weasel. In other words, the changes for trial 2 and 10 probably not going to help reach what we're concerned with. A major concern is hotspots, and there are many, many examples. P53, which is damage of P53, of course, is a major cause of colon and other cancers. This happens to be P53 gene in colon cancer, and we can see that the vast majority of mutations, and we're looking at several thousand evaluations, almost all the mutations are in 4 different areas, and several areas had no mutations. And so this is a major reason why cancer occurs, because they're not random. They're in specific areas. And we're looking at the cause of a cancer in a particular patient. We look at the mutation that caused cancer in that patient, and more they're able to use this information to help treat patients. Now, these are called hotspots. And here again, other studies have found not just genes that cause cancer, but in other areas as well. You can see that there are many hotspots. E. coli, 199, number 199, that is where, in this case, about 90%, 90 some percent of all mutations occur. And one study found only 2 mutations in the hotspots. 2 mutations accounted for 94.4% of all mutations in the 319 they identified. So the problem is hotspots will keep changing, and therefore they will cause a problem and then reverse, which sometimes we get a mutation which actually can fix a problem. And that's because the mutation occurs again in the hotspots and goes back. Now, we know that conversion to the other bases is not the same. Conversion to thymine, for example, 58% of conversions occur. The guanine, 14%. So you're far more likely to end up with a conversion to thymine than guanine. Conversion to cytosine, 23%, adenine, 5%. So what's going to happen? You're going to build up more and more thymines, which causes, in most cases, deterioration of the genome than you are any of the other base pairs. Change charge and polarity is important as well. We can't just look at the change into specific, in this case, base pairs. What we have to look at is for the protein to function, to fold, it has to have certain nonpolar uncharged, polar uncharged, borderline polar nonpolar charged, polar positively charged, which are critical, and polar negatively charged. And so mutation too often results in this change, and that distorts the protein, and we end up with a problem such as cancer. And so the evolution account is valid. We can see that a change, in case of IS, it's IA, and the changes from a functional gene to a non-functional gene is far more likely than the opposite. Linsky did a well-known study, started in 1988, involved 12 populations of Escherichia coli, and he had 75,000 generations. And the culture medium had not much glucose, but a lot of citrate. So what happened was when the microbes consumed glucose, they would continue to grow only if they could evolve some way, or in other words, a mutation could occur to allow them to utilize citrate as a source of nutrition. Of 12 culture lines, bacteria developed the capacity for metabolizing citrate as an energy source under aerobatic conditions. And this was due, they think, to 2 mutations. So the likely scenario, they concluded, was mutations jam their regulation so that bacteria produce citrate transporter regardless of the oxidative state of the bacteria's environment. So yeah, it in this situation turned out to be positive, but as a whole caused a number of problems. And of course, it took, 31,500 generations to get this single change. And the example is comparable to having a light that switches on when the sun goes down. Sensor detects the lack of light and turns the light on. A fault in the sensor then. Sensor doesn't work, so the light may be on, therefore, all the time. Another possibility is an existing transporter gene, like the one that normally takes up tartrate, mutated so loss of specificity. And this is, from my experience, quite common in mutations, how they produce a benefit, they lose specificity. And such loss is an expected outcome of random mutations. And so in this case, you would conclude, yeah, probably due to random mutation. And other research has been done on this, but I'll move on. One problem we have in understanding the genetic system is the interactome. The interactome is where we have proteins and genes which interact with hundreds or many hundreds of other genes. And so when one gene becomes damaged or one protein doesn't work anymore, what we have is it may be a beneficial for that one protein. It may produce a beneficial effect, but it all too often has negative or potentially negative effects to other proteins because the host system is heavily tied together. Now, there's not a lot of yellow lines here because they're still working on this interactome. But the one gene in the center, towards the center, you can see NEFL, I guess. That has a lot of connecting points as others don't. The reason they don't is not because they're not there, but because they're still working on this. Takes a lot of work. Lethal problems. I, as once was an evolutionist, an atheist, and what converted me and lots of other people, I have a whole book on this, to the creation worldview was studying evolution itself and realizing that it couldn't occur. Just a few brief examples. Prokaryote to eukaryotes, endosymbiosis fails. I did a whole paper on that. The evolution of sex. Evolutionists baffle as how in the world sex could evolve. Obviously, the male and female reproductive organs must be compatible. Otherwise, you're going to have extinction. So to go from asexual to sexual has always baffled. As what Darwin said, it's a great mystery. And evolution of life, there are 3 examples: horses, humans, and whales. And in all 3 of these, I've done papers or books on the problems, the claims between the evolution of horses, humans, and whales. So the evidence, I concluded, and many others have as well, against evolution is overwhelming. It's somewhat embarrassing that indeed so many people in science throughout the world accept the evolutionary viewpoint when there are so many, many problems. I've got over 2,000 papers now that I delineate many of these problems. And some people say, well, you read creationist literature and you'll get these ideas. I rarely actually read creationist literature. I read primarily evolutionary literature. The evolutionary literature is really quite clear if you dig into it on the conclusions. And so my conclusion from reading evolutionary literature was there is a major problem in all these major steps for evolution. And there you can see Darwin's secret sex problem, where Dr. Smith documented the problems there. Here's one book of many that I have written called Fossil Forensics, where I show the fossil record is woefully inadequate and the few claims that are made are problematic. So more complexity. The effect of genes can vary greatly depending upon which parent it's inherited from. So a gene inherited from mom produces one effect, from dad produces another effect. The point of this is that the complexity is enormous. And this is why dealing with cancer causation is so difficult, because there are so many issues that are involved. And the body parts of a car, before the car functions, you have to have all the parts. And for so many structures, you have to have all the parts before it will function. And a Great Dane and a Chihuahua, and this is a question I want to ask Zach. These two, I know there's a problem. You got to have the male has to be, or the female, pardon me, has to be the larger dog. But in my class, I said a Great Dane and a Chihuahua cannot mate. They cannot produce offspring, period. Well, I had a student, bright student, raise his hand and say, I'm a dog breeder. And I had a Great Dane and a Chihuahua, and they did interbreed and they produced a viable offspring. And I was amazed. And he said, yeah, we were too. This was not deliberate. We did not do this purposely.
1 minute left, Jerry. 1 minute.
It just happened. And so there you got a Great Dane and a Chihuahua mix. So they do mix. And I find it amazing that you have like 2 different books. You can put them together, and yet the resultant book makes sense. And I don't understand how that could happen. And I've asked many people, and they say they don't know. But Zach is very bright, obviously, from his presentation. Maybe he could help me figure out how indeed you can get mixes, and there are, of course, thousands and thousands. His field is population genetics, and of course, that is where evolution is obviously true. So he's got the facts there, but of course, creationists call that microevolution. That's not what we're concerned about. But anyways, that's quite amazing how this level of evolution could occur. And that's it.
Dr. Jerry Bergman, thank you very much for that 18-minute opening statement. Gentlemen, that concludes our opening statements for tonight's debate. I appreciate the time you both took to construct those opening statements. We've got plenty of points now on the table to engage and rebut in a rebuttal. Rebuttal for this debate is 10 minutes. So I will reset the timer and hand it back to Dr. Zach Hancock. Zach, whenever you're ready, you have 10 minutes for a rebuttal. The floor is yours.
Okay, thank you so much. That was interesting. I want to be sure that somebody clips what Jerry said there at the end, that evolution is obviously true. That was one of the last things he said there, so I'm going to be sure to clip that. I want to start off by talking about, we'll sort of work through some of Jerry's presentation here. And the first thing that he started with was that beneficial mutations break stuff, right? That's sort of the first thing that he began with. And I think that I spent a lot of time in my opening statement showing lots of mutations that don't break anything. In fact, I made sure that every single mutation that I showed you were mutations that don't break things, but that build new things, right? Also, we're doing this kind of conflation of what beneficial is. Beneficial is about fitness effects. Well, what we really care about is, can we build new things? So let's be sure that we're keeping those two things different from one another. I also want to give Jerry a bit of a shout out here. He's the first creationist thus far that has actually addressed, or at least said, our paper live on air. So I'm pretty excited that somebody has acknowledged our paper. Whether or not he's read it or we're going to discuss it is maybe another question. He spent a little bit more time talking about innumerable forms in the fossil record. This is really a discussion about mutations. That's what the arrival of the fittest is all about. I can't infer mutations from rocks, so I'm going to basically leave that one be. I do want to, however, address, he showed a picture of a book about monkey to man evolution, picture book thing, in which he said that the person claimed that most fossils are fakes. Now, that's one I can't let stand, because that's an accusation. And we need to really be careful when we start accusing people of lying and faking things. I think that when we have these scientific discussions, we should never go to, well, it's a fake or they're lying to you. I think that that's something that needs to be brought and made very seriously, and you better have some very good evidence for that. That's not a throwaway line that we can just say. He then showed this March of Progress quote. And he attributed this to Alexander Worth, that basically humans and the March of Progress had shown the humans on top of everything, and this has been such a damaging way of viewing life. What Worth was not talking about evolution there. He was talking about thinking that humans are on top of everything, which is what creationists believe, right? So really, he was warning more against thinking that humans are exceptional and that we should dominate the planet, and that we should really think about ourselves as integral to the planet. Not that there's this march of progress towards better and better humans, that's a false way of thinking about evolution. And I've really tried to promote this in a lot of my work as well, but that's what Worth was talking about, not that evolution itself is some problem. The next thing that Jerry talked about was human-chimp genome similarity stuff. Whether it's 98% or 85%, to me, is largely irrelevant. So if you want to say that humans and chimps are only 85% related to one another, then that means that two humans must be greater than 8 or 9% distant from one another. Like, you have to include things like lions and tigers that you would assume are share a common ancestor. They end up being greater than 20 to 25% different from one another, and you assume that they share a common ancestor. So I don't really understand why we're so hung up on these specific numbers, because in the evolutionary framework, we're all related. We're related to banana plants, right? And they're only 50% of our genome. So the specific number tells us about how long ago we diverged from chimps. It doesn't tell us anything about whether or not we are related to chimps. That's a fact, because we all share the same heritable material. Right, that's a completely different kind of argument. So the human-chimp genome similarity thing, to me, is a red herring. The next thing that he went to is that the vast majority of mutations are deleterious, and that's a problem. It's not a problem. It's, in fact, exactly what is expected, and it's what is necessary. If the vast majority, or if even 50% of all mutations were beneficial, if they were like half and half, then because beneficial mutations are favored by selection and they necessarily increase population growth rates, then they would fix so quickly that the population would just balloon in size. We would be neck deep in elephants if 50% of all mutations were beneficial. If most mutations have to be deleterious, otherwise you would have this sort of runaway selection and populations would explode in size. It's very important that only a minority of mutations are beneficial, and that's how you can maintain sort of a fitness equilibrium. And we can talk about that in more technical detail if Jerry's interested in sort of the open discussion. Jerry really likes to talk about this, and he thinks it's like a weasel thing. This is sort of a thing that Dawkins did. What Dawkins is trying to show here, and I think what's often missed, is simply that random mutation plus selection can generate a target. right? And I showed this empirically in the bacteria example, right? When I showed the fitness landscapes, that there were some high peaks of fitness, and that if you randomly mutate the bacteria, some of them will find those peaks of fitness. That's all that's being done here, and that's what the methinks like a weasel is supposed to represent. Jerry thinks that somehow hotspots undermine that. I really don't understand that argument, because hotspots are things that we've known about for a very long time. In my field of phylogenetics, what we can actually control for hotspots. All you do is you add a little extra term when you're modeling the substitution rates, and this extra term, we call it the gamma distribution term. And what it does is it draws higher values across different sites. And this enables you to effectively model hotspots across genomes when you're reconstructing relationships, when you're measuring rates of molecular evolution. Like, these are fundamental things, the models of which go back to the 1960s and 70s. Like, some of Kimura's first work was actually building substitution models, right? So around things like hotspots, around things like biases and mutation probability, so the difference between transition and transversion rates. The OG substitution models, like the Jukes-Cantor model, the Tamura May 83 model, like those kinds of models were all about modeling exactly what Bergman here was talking about. So we are aware of these things and we account for them all the time. He brought up the change in charge and polarity for whenever you're going to change a protein, you're also changing the charge, the polarity, those sorts of things. That's really why I tried to focus on showing these protein pairs that have 17 amino acid differences. They have differences in their conformations, differences in their hydrostatic interactions, right? So to change from one to another involves changes in polarity. You are fundamentally changing conformations and the interactions between those two things. And we showed that there's a step-by-step Darwinian way to pull that off, and you never lose protein function along the way. So this is a thing that we can very clearly see does in fact work. He talked a bit about the interactome, and this is a really fun topic, and we could get really, really deep into the weeds on this. And the idea about pleiotropy and that all genes are kind of interacting together. In recent years, we've come to call this the omnigenic model. Jonathan Pritchard came up with this. And what's really fun about this is it is an empirical verification of a model put forward in 1918 by R.A. Fisher called the infinitesimal model. So basically, the entire field of quantitative genetics was built on the idea that all genes have multiple interactions and they're effectively are pleiotropic. And what he showed is that if a trait that you possess is impacted by every single gene in some little way, right? Those that are closer in the interactome impact larger. Then as you get further and further away in the nodes, the impact becomes smaller. If that's the case, then he wrote out the math in 1918 of how selection should behave on those things. That is where we get things like the breeder's equation. That's where we get all of quantitative genetics comes from this basic thing. And that's why we have the livestock, the crops, all the things that we have around us, is because we assumed a very complex interactome from the beginning. That's where our math comes from. So again, we can get really into the weeds of that if we want to, but I just want to point out that like that's a fundamental part of quantitative genetics. He then spent a little bit of time talking about the evolution of sex. That's maybe a little outside of the scope of Arrival of the Fittest. We can talk about it if you're interested. But we would need to pin down, are you talking about the evolution of meiosis? Are you talking about the evolution of daiichi, like separate sexes? Are you talking about just the specialization of gametes, like an isogamy? Like what exactly do we mean here? Well, we'd have to really sort of nail that down. Lastly, I want to point out that complexity emerges passively in evolution. That in fact, selection often tries to keep things simple and efficient. And it's not until you get eukaryotes, large-bodied vertebrates, plants, these big things with small population sizes, that things like drift and mutation pressure get strong enough that you can actually passively emerge complexity. And you passively emerge complexity because of things like gene duplication, things like subfunctionalization, these sorts of jargony terms that I'd be very happy to talk through in greater detail. But I've only got 10 seconds left, so I will leave it at there. Thank you.
Dr. Zach Hancock, thank you very much for that first rebuttal of 10 minutes. To the audience, I appreciate all of the interaction and engagement so far on such a great topic. And I'm all caught up on your questions. So again, if you have a question or a challenge for our guests, feel free to send those in, and we look forward to interacting with those. Dr. Jerry Bergman, I have reset the timer, and you also have 10 minutes for a rebuttal. I'll start the timer now. Go ahead.
Okay, my book here, which haven't read the whole thing yet. I'm working on it, but I've read reviews and I've read the first couple of chapters. I don't think he used the term fraud. He used the term that a lot of these claims are inactive, or they're not correct. That indeed a certain fossil was claimed to be Australopithecus afarensis, and there are problems in that conclusion. So he goes into great detail justifying his conclusions, but a rejection of the progression. And by the way, there's many researchers now have done 2 or 3 papers on that. They reject that, but I have to wonder, how did evolution occur? Because the progression is so effective, it converted many people to the evolutionary worldview. One glance and one's convinced, and yet it's largely being rejected and replaced by other worldviews. And so I find that intriguing that they don't seem to realize that they got a problem in whole evolutionary theory. One concern I have with Zach's presentation is that he finds one organism has 3 similar genes or 3 identical genes. He assumes that as a result of gene duplication. It may be, but it may not be the result of genetic duplication. I've done a couple of papers on genetic duplication, and I find a number of problems with it. Namely, when you have a duplication, it can, such as in certain disease like Huntington's chorea and other examples, can cause a problem. So gene duplication can indeed cause a problem. Also, the idea is that the one gene continues to function, the other gene mutates and evolves into another gene or another function. Well, the problem is that assume that both genes have about the same likelihood of mutating. So how do we know that the one will continue to function and the other then will evolve into another gene? That's problematic in many cases. That's an assumption that's problematic, and others have less faith in the gene mutation idea. And so a problem I have too is that evolutionists tend to see evolution in what they look at. They've got these evolution glasses, and all too often that is distorted. And of course, the best example is this book right here, where you get some fragments, some fossil fragments, and you put them together and you end up with a progression. And this often resulted in distortion, partially because you see what we want to see. You see parts of a skull, and it appears that it could be a organism in between monkeys and humans, and therefore you label it a missing link. And that this is a major focus of this book, is to question that a lot of these, they saw evidence that really wasn't there when one looks at it in more detail. I think the primary reason that the progression was rejected is because we have more and more fossils. More fossils do not show the progression. They show groups of organisms. They show 4 or 5 families. And that is a major problem which he looked at. But in my work with the medical school, mutation, we're talking about they're problematic. We all got our Geiger counters, and we all made sure that the environment we were in had the lowest level of radiation, ionizing radiation as possible. And let's see if we have any here in the room. A little bit. It's called background radiation. And obviously you can't get it to 0, but you try to get it to as low as possible. And therefore, it's a concern. And sexual reproduction, we're concerned about a male and female animal which is able to sexually reproduce. I'm not concerned about, at this point, meiosis, mitosis, and so on. We're concerned about, can animal A, a male, animal B, a female, sexually reproduce? And that is a major problem Darwin had, and it's a major problem that evolutionists have today. They admit it's baffling, and it is a major problem. If you think about how they could evolve and reproduce during the entire time of evolution, from asexual to sexual, it's baffling. And this is just one of many problems which evolutionists admit are a big problem. and they're not focused on it. There are books out there that do focus to some degree on this, but they propose theories as to how it could occur, how sexual reproduction could occur, but so far none has received wide acceptance in even the evolutionary community, and so therefore they are problematic. And when I mention many of the books I read most are evolution, why can I read so many and see creation in them? Because so many come up with conclusions, and you realize what is the foundation of these conclusions? What is the evidence for their conclusions? When you look beyond what is stated, when you analyze the conclusions made, you find out that so many of the conclusions are not viable. They're not appropriate. And I find when you see the world through evolution glasses, you see so many things that are really not what they seem to be when you're wearing those glasses. And so when I read these books on evolution, I don't have a pair of evolution glasses. So I have no glasses at all. So I tend to see the material for what it is, and I can see it often supports the creation or intelligent design worldview. And therefore, looking beyond it. And I can see in reading papers that so many, they're just trying so hard to make this fine to support evolution. And all too often they can't. And they may admit that, but more research and we'll be able to do that in the future. Or this is a question which we still have to answer, and we are going to look at in more detail. But the more research that's done in the area of evolution, the more problems actually that arise. And I've often said that evolution will be disproved, but it will be disproved by the evolutionists themselves. And this is why when I got this book and several others, this is exactly what has happened. And therefore, when you look at the whole picture, you find it doesn't support the evolutionary worldview. And by the way, there are many, many people, I mentioned I did a whole book on this, that come to the creator by evaluating the scientific evidence. And so science can be the doorway to the creator. All too often, evolution is the doorway to atheism. And the opposite, though, can be true as well. And so when I mention this to others,
30 seconds left, Jerry.
When I mention this to others, so often I have people say, yeah, I took the same route. So it's not uncommon at all. In fact, the more educated, effective creationists came from an evolutionary background. And so that's a few comments.
Dr. Jerry Bergman, thank you very much for that 10-minute rebuttal. Gentlemen, we are moving along smoothly. That concludes our opening statements of 18 minutes and our rebuttals of 10. Clearly, we have plenty of points and arguments for our positions tonight on the table to engage in a discussion. Now, discussion-wise, we have 45 minutes on the clock, but this will be less strict. It's not a strict cross-exam. We're going to try and keep it more free-flowing and organic. And what I'd like to do, though, is to do our best to keep it to one point or one topic at a time. That way there won't be too many points on the table to address at once, and that might help it to keep a little to keep it a little more focused. Now, what I'd like to do, since Dr. Bergman just ended with his rebuttal, is Dr. Hancock. Let's give you the opportunity to kick us off in this discussion by picking a point or a topic that you'd like to go back and forth on. Go ahead.
Yeah, okay, cool. So I'm going to get sucked into this evolution of sex thing. I see it already. I'm going to get sucked into it. So I didn't want to do it, but we're going to talk about it. So that's I think going to be the first thing. So what I kind of want to start off with is that the big contention actually is not about the evolution of daiichi, which is separate sexes. The contention is about the origin of meiosis. So if you're concerned about the origin of male and female, that's actually pretty easy to talk through. So are you aware of hermaphroditism? So the idea that an organism possesses both male and female gametes and tissues for producing male and female gametes.
Of course, yeah.
Yeah.
Very familiar with it, yeah.
Yeah, so most basal organisms, when we think about like the origin of animals, the origin of plants, organisms that are beginning to be multicellular, these organisms were all hermaphroditic. So what they were doing was specializing the production post-meiosis of different kinds of tissues for developing sperm and egg, right? They didn't evolve males with no female organs and females with no male organs. That's not what happened. It all specialized in one individual or in one group of individuals, and then you had mutations that would shut off development of one or the other one. So you would get individuals born that were lacking both but had one. And now this individual that's either male or female, either has the production of male gametes or female gametes, can still reproduce with anyone in the population because all the other individuals are hermaphroditic. And I can give you some actual examples of living organisms that do exactly this. C. elegans does this, right? C. elegans is hermaphroditic, but will occasionally produce strictly males, right? And so the idea is that you get these mutations that will shut off the sex system of one or the other one. And then when you got hermaphrodites and the single sex system, those hermaphrodites are now expending a lot more energy to build that extra sex system than the other ones. So there's now a pressure for them to shut off the alternative form. And this is where you get like the evolution of sex chromosomes, right? And so that's how separate sexes evolve. You don't start off with like two different individuals of separate sexes. Does that make sense?
Oh yeah, I say that's a story, and it's a story which is plausible, I guess. But again, it's a story, and I know hermaphrodite animals. You can build up this story, but there are many problems with it, which several researchers that I've read go into it and explain why this is not very viable. The story is not very viable, and especially since you talk about the enormous complexity of male and female gonads and the sexual reproduction system, which involves not only the gonads, but also the mind, the sexual drive as according to the brain development, which is...
Well, no. We're talking about like flatworms, right? Like the origin of this, the origin of separate sexes would have been back to like bilaterians. Like flatworms don't have brains. The organisms we'd be talking about, it's literally just the differentiation of sex gametes. It's not about all the complexities of like male and female humans or even male and female vertebrates, right? This would have been, males and females would have originated much, much, much more ancient in time than that.
Yeah. I can understand with platyhelminthes and some of these flatworms. That's intriguing and it's interesting to get into, but there's an enormous gap between that behavior and the behavior we see in mammals, for example. And extrapolating that from one to the other is, I think, problematic. Do we see much evidence of that in the fossil record? Well, yeah, the saw parts don't preserve very well, so it's not going to be there. So indeed, one can come up with reasons for it not being there. And these reasons may sound appropriate, but indeed, the fact is, are they true? And again, we're building stories.
Yeah. So again, I mean, the question was is there a plausible explanation for this? And is there organisms today that can provide evidence for it given their current sex systems? And there are, right? So we have a plausible explanation for how it can occur. And then we look at, are there systems that exist in nature that follow this? And I gave one example, and that's C. elegans. And there's lots of other examples as well of like new sex chromosomes that are emerging within populations that would also lend evidence to the idea that these sex chromosomes emerge mostly by like large inversions and things like that. So just in Drosophila, for example, since we have kept them in labs, since we've sort of domesticated them, a neo-Y chromosome has evolved. This is a brand new sex chromosome that has evolved just in the experimental populations of Drosophila. We have beetles with brand new sex chromosomes in natural populations as well that we've observed. So it's a story insofar as it's an explanation, and then I'm relying on observable evidence, like data that can support that explanation. So it's not just a story without any sort of like support for it.
Okay, my response is, yeah, this is all interesting, and but flatworms are doing pretty well as to how they reproduce, doing very well, and to bridge flatworms to mammals is quite a gap, and it's not nearly as easy as you imply. And again, if it's doing flatworms, there's doing, of course, I know there may be problems in some areas thanks to humans, but on the other hand, flatworms are reproduced quite effectively. And therefore, it's quite a gap to go from flatworms, which are, again, around today, and then to assume, well, we evolved from flatworms, or we evolved way, way back from flatworms that back in the evolutionary history, and flatworms eventually became mammals and eventually became, of course, primates and humans. So that's a...
Jerry, if I could maybe add a question, what would be, and then Zach, perhaps you can interact with it, what would be a couple problems scientifically with what Zach is presenting for the origin of sex?
Well, he's taking specific animals and extrapolating that to a series of hypothetical changes, which brought us to where we're at today with male and females. And when I mentioned the brain, of course, obviously, flatworms don't have a very elaborate brain. I guess they have neurons primarily, but it's quite a jump to go from animals which are still around and assume that they were our evolutionary ancestors, which from them our sexual reproduction systems evolved. And it's not for mammals, it's not just the sexual reproduction system, but it's also for birds making nests, laying eggs, and nurturing the young. So it's a very complex system, which flatworms don't produce eggs, they don't nourish the young. So there's quite a difference. So again, we're taking animals and theorizing from them. And again, these are, as Gould, this is Gould's expression, by the way, we're creating a just so story. And all too often we are creating, as Gould recognized, Stephen Jay Gould recognized, these are just so stories. And
so what they are hypotheses, right? So if we want to know something that happened a long time ago, like if we ask a big question like how did separate sexes originate, right? Separate sexes exist across life, right? Like the evolution of separate sex, one happened many times, but it's widespread. And so we need to then make a hypothesis about that, like how did that process occur? That's the first step. Now that necessarily is a question, or is necessarily a statement that sounds like a story. So you tell, this is my hypothesis, and then you say, okay, can I support that hypothesis? And so what that hypothesis implies is that there must be organisms that are hermaphroditic that are also capable of producing separate sexes. And then we should also find mutations that drive those separate sexes and fix them into place in natural populations if our hypothesis is true. Those are things we should expect to see because that's the natural process that must entail. So then we go out into nature because I can't look 2 billion years ago. Like I have to rely on what's alive today. I go out into nature and I see if I can find organisms that fit this explanation. And if I can, then I can point out, look, there are natural examples that match what my hypothesis predicts should have been able to occur. Now, does that mean that that's exactly how it happened or imply that flatworms that are alive today are our ancestor? Not at all. It's about support for a hypothesis that originally you claimed was impossible and some gigantic problem for evolution. And I don't think it is. Again, the problem for evolution is about meiosis, not about the origin of separate sexes. I hope that's clear. I don't want to spend too terrible much time. I'd like to talk about genes and mutations, but I just wanted to make that as clear as possible.
Yes, let's do this, gentlemen. I appreciate the back and forth on this topic. Jerry, let's throw it to you for a quick response. Then Zach, let's throw it back to you to pick another question or topic, perhaps related to mutations and the arrival of the fittest. So Jerry, feel free to give a quick response to that.
Okay, I've done a lot of work on fossil record, and it's quite clear the fossil record that we're baffled as to where so many organisms came from. Horses, dogs, I know from the wolf, but where the wolf come from. And so many animals, there's just no sufficient fossil record, no good fossil record. In fact, most of the fossil record for most organisms are very, very poor. And so we're talking about evolution from some asexual to sexual organism. We don't even have evidence for the evolution of birds, of most mammals, of every organism I picked. I do detail what evidence we have for the evolution of these organisms. And it's just, there basically is none. There's lots of theories, lots of hypotheses, like whales evolved from some dog-like creature. That's a theory that they've developed because they're trying to find an explanation for the origin of whales. Well, there are serious problems with that from dogs to whale hypothesis, from a mammal or a terrestrial animal to a marine animal, a whale. And these problems are admitted in the literature, but the explanation is, well, we keep looking. Fossils are there, we just have to keep looking for fossils. Or the fossils would be so old, not many have survived, because most fossils, of course, don't survive in the fossil record, and therefore that's why we can't find them. But when you see the same thing over and over and over, the same excuse about every animal that I looked at, fish, birds, primates, etc., you see the same problem. And you see that you have to have...
It seems the fossil record's a little far from the origin of separate sexes. And so like, I also don't want to get dragged into discussions about the origin of species in the fossil record. So if we could maybe move to talking about the mutational process.
Yeah, Zach, let's throw it back to you. Feel free to pick the next topic or question for you guys to engage. So specifically on the question. Go ahead.
Maybe we can talk about duplicated genes. One of the things that you said in the rebuttal was that there's a problem with gene duplications. And in particular, that when duplicated genes emerge, they can be problematic. Could you perhaps elaborate on that?
Well, many examples in disease, Hancock Curry, of course, the most well-known example, but we know that duplication of genes produces too much of something, which causes a problem. And I did, again, many years ago, I did a paper on the problems of duplication of genes idea. And a major issue was indeed that you end up with production of too much of something, too much of some protein, for example, in Huntington's chorea. And,
so if we can, let's take like ribosomal genes. I think are a really great example. Ribosomal genes in humans have a really large variation. So just between the three of us, we can differ in the number of ribosomal genes we have from 100 at the low end to over 500 at the high end. Right, so the number of RNA genes are wildly different. Ribosomal RNA genes are wildly different, right? We also can differ in just our total genome size, right? So like between all of us, we can have 100 million base pair differences in our genomes. And so what this implies is that duplications must happen all the time. And in fact, we know they do. They happen as common as single base pair mutations. And if duplications were always super harmful, then that would seem to suggest that we wouldn't have such variation in the number of like ribosomal genes. But that doesn't mean that they're not occasionally harmful, but I think that they're probably far more often neutral than what you seem to imply that they are.
Okay, I understand that. Yeah, I'm not saying they're always harmful. I'm saying there are many situations where they're harmful. And of course, in the medical field, you're interested in mutational changes or changes, period, which can cause a problem. And over and over in studying this area, gene duplication was maybe overgeneralized, but it was generalized as a problem. And yeah, there are examples where massive duplication can occur, as you mentioned, in ribosomes, and there is not a problem. But on the other hand, in so many situations, there is a problem. And therefore, I think that...
Yeah. So sometimes there's a problem, sometimes there's not a problem, right? So let's take the instances in which there's not a problem and you get a duplicate gene. Can that daughter copy or the parent copy, either one, since they're redundant, can it mutate and acquire a novel function that its parent copy didn't have? Do you think there's good evidence of that?
Oh, well, that's possible. But I wonder if there's good evidence of that. It's easy to look at an organism and say, well, these two genes are similar, so therefore the gene must have duplicated, and therefore this gene continued doing what it's supposed to, and this other gene evolved into doing some other function in the cell. It's easy to look at that, but how do we know indeed that that's actually what happened? We can't go back in history, and I guess we could if we could do analysis of a number of organisms and find that this organism for many years had no duplication of this gene, and then all of a sudden here we can pinpoint the organism that did have the duplication, and we can pinpoint the changes made so that indeed one gene continued to do what it's supposed to, and the other gene evolved in doing something else. If that could be done, I would say, yeah, that's convincing. But otherwise, I would say if we have one gene, a similar gene next to it, which has a different function, to assume that this is a result of gene duplication, and the gene that was duplicated evolved into some other function. Again, though, I look at over and over again, the major problem you have in mutations is the vast majority are problematic, either near neutral or
before we get there, so it seems like you have a problem with the concept of inference, right? That if you don't literally see it happen, then you can't infer that it did happen. This is right, like the crime scene problem. Like if you don't see a crime happen, can you still infer that a crime happened given the evidence available to you?
Oh yeah, you can infer it, but just
right.
make
So
sure
if I have then two species, like two Drosophila species, Melanogaster and Pseudoobscura, I assume they're all fruit flies. I assume we accept that they had a common ancestor, right? And so we can look at the exact same gene in the two of them, align them, see that they're the same gene. We can do functional assays and they do the same thing. But then it turns out in one of those, there's another copy, right? And that copy's younger. And we know that it's younger because it's less diverged from the fly that it's in than it is from the sister copy. That's the only way that can happen. That's the process that we're inferring there. And if we see that daughter copy turns out to do different things in the cell, then the inference then is that it emerged via duplication and has acquired novel functions. Now, that's because we know duplications can occur. We have literally seen duplications occur, and we see mutations within these duplicates occur, right? So now all we're doing is taking what we observe, and then we're inferring based on the patterns that we see in nature, right? Which is the same thing you would do in a crime scene, right? You know that crimes can occur, and you can recreate what had happened based on the evidence available.
Yeah, and just state you're inferring it and give the reasons why, etc. But I find all too often that's not how it's stated in the literature. It's stated that indeed this is what happened. And we're not inferring this to happen. This is what happened, period. And therefore I have a problem with that. And again, I have a problem with the whole mutation idea because as we agree, the vast majority, 99% or 98% are harmful and cause problems.
Yeah.
And when I was at the medical school, it was indeed stressed that these are a problem. You want to protect patients from radiation, which could cause ionizing radiation, which could cause mutations. And it should be ideally zero. It's not, but it should be. And so therefore the focus is, and I could see that importance in the medical area. I could see the importance of mutations are not good. If even a small percent were beneficial, maybe we'd be less paranoid about exposing ourselves to radiation. But as was made clear when I was certified to work with radioactive materials, as was made clear, radiation is a problem and zero dose is ideal. You can't reach that, but it should be as low as possible. And therefore, it's going to cause a problem sooner or later, and therefore you need to reduce the rate as low as possible. That's why.
So do you think it's possible to infer that a gene duplication has occurred and then that duplicate has acquired a new function? Do you think that that's possible in the same way that we infer like what has happened at a crime scene? Do you think that that's possible?
Oh yeah. No problem inferring that. Yeah, that's
okay. So if we can infer that happens with let's say some level of statistical certainty, right? It's a confidence interval kind of thing. If we can infer that it happens, and we have inferred that it has happened thousands of times, right? Like the examples that I gave in my opening were not just one time or just a couple of times. It was thousands of new genes, both regulatory genes as well as protein coding genes. So do you not think that that's pretty strong evidence that the gene duplication process is capable of generating novel things if we're not just seeing it one or two times, but we're able to infer it thousands of times within organisms that I think we would all agree, all creationists would agree, share a common ancestor?
Yeah, it's possible, but I should have, I guess, reviewed my paper before I got on here. I didn't know that you were going to spend so much time in gene duplication, but I know in doing my paper, which is probably 10 years old or 8 years old, I know a lot of literature indicates that it is often a problem, and it's not a viable source of new genes. Didn't say it was never, said it's not in a large number of cases. And I don't remember the data that was given, but the literature was pretty clear that this is not the answer, not the be-all for evolution. And so that's...
Yeah, I mean, that feels a little difficult to accept, given that I gave a single study from over 10 years ago, right, that found 4,000 brand new regulatory genes in one species. But so maybe we can move and we can talk about the deleterious mutations, because that which is what you were bringing up. So if you want to talk, we can talk about the proportion of deleterious mutations. It is definitely the case that most mutations are either neutral or deleterious. Like that's a fact of nature. I think all evolutionary biologists not only accept that, but we say that.
I would say near neutral. I would say near neutral.
Well, yeah, we can split hairs. Near neutral, if you like. They all probably have some effect, right? And the majority, when we start talking about near neutrality, it becomes complicated, because then it depends on the organism you're talking about, will dictate how many of those mutations are near neutral, because near neutrality is a measure of drift, and it's a measure of the effects, the differences between drift and selection, right? So that becomes a little bit more complicated when we go that route. But in effect, the fact that the vast majority of mutations are deleterious is actually not a problem at all for adaptive evolution. So long as you have just a tiny fraction of beneficial mutations, you can still get adaptive evolution. Now, you showed our paper at the very beginning of your talk, and that's what our paper shows, right? Is that if you have a mutational distribution in which the vast majority, 99%, are, as you said, near neutral, right? So they're actually technically slightly deleterious. 99% are slightly deleterious, or they're harmful. Like we have like an exponential distribution, and only 1% of them have any beneficial effects. The vast majority are less than 1% benefit, right? So it's a tiny benefit, even when they are beneficial. And what we found is consistent adaptation, that is consistent population growth in all 5 replicates that we looked at. So how do you explain that if the preponderance of deleterious mutations is such a problem?
Well, I'm reminded of, again, my coursework at the medical school, and maybe we shouldn't worry so much about being exposed to ionizing radiation. Maybe we shouldn't be so cautious walking around wearing lead suits and all the accoutrements we had to work with to reduce the level of mutation. But again, it was stressed that they're all harmful, got a problem, and therefore you got to have your Geiger counter, you got to check the background radiation, got to be as low as possible. And if it's too high in certain areas, like certain gases, ionizing radiation comes in through certain gases, and homes, they used to be a problem, and that needs to be taken care of. And ideally, you need to get it as low as possible. So, and again,
so we're in agreement here. Like, I agree that 99%, probably even a little more of that, of mutations are deleterious. I'll accept that. I will give you that number. That's not the debate. The debate is that if a tiny number of them are beneficial, you can still get rapid adaptive evolution. Because again, selection is what's driving adaptive evolution here. The vast majority of mutations are lost, right? Because if they're deleterious, selection is acting against them. And if they're near neutral, then drift removes them. The vast majority of them never go anywhere. And this is why just a tiny portion of beneficial mutations can still lead to adaptive evolution. Now, again, we showed that in the paper, but we weren't like the first people to do that, right? Like, this goes back to R.A. Fisher. It's not like we did something new. But to me, the question still is if the preponderance of deleterious mutations is such a problem, why do we consistently see adaptive evolution in these models that take into account that preponderance? And as well as, why do we consistently see adaptive evolution in nature? When you go out into nature and you measure how much additive genetic variance, that is the amount of genetic variance that can contribute to adaptation, when you see that in nature, there's a lot of it, right? So if what you're arguing is true, those two things shouldn't be true. We shouldn't see this in the models, and we shouldn't see it in nature, and yet we do.
If so many are deleterious, though, you're going to have a lot of natural selection. We're removing these deleterious mutations, awful lot. And yet fortunately, most animals don't die of mutations at a young age. Most survive to be whatever their normal age is. We've had several dogs, and they have all survived to the normal life expectancy for a dog. In my family, most of us survive the normal life expectancy for humans. And therefore, if you had some, but again, it's like Dawkins talked about the Mount Improbable. You have one step forward, 99 backwards. You're never going to get to the top of the mountain. You'll always be going backwards. And therefore, with a so high level of deleterious mutations, you're always going to be going backward. Now, I know they're limited through natural selection. And that's one function natural selection clearly has. So it's going to reduce the number of deleterious mutations. And that's, of course, what you see in the hospital situation when you have a child that has certain deleterious mutations, has certain diseases, and dies. That's not going to be passed on to the future offspring. So indeed, that is one important function of natural selection, is to reduce the mutational load, and it does serve a very important function, and that's one function it does serve. So that's not the problem. But when you have so many deleterious mutations and one beneficial mutations, that's just not going to survive. It's going to be selected out. And therefore, we're going to have a lot of selection among animals. And
I think you have a pretty bleak view of how these things get selected out, right? So the mutation that's deleterious doesn't have to kill you, right? It's a probability statement. It's about how much does it reduce your chance of survival or reproduction, and sometimes it's even relative. So I think we're thinking about deleterious mutations in a lot harsher way than what they're actually are. So like, if the survival probability reduces your chance by just a tiny amount, then maybe only one individual with that mutation doesn't have offspring, but the vast majority of people might still. And over time, selection will still weed that out, right? So it doesn't have to be some like horrible genetic disease kind of thing. That's not really what we're thinking about when we're thinking about deleterious mutations. But again, I've shown bacteria that have been able to find peaks of fitness by just being subjected by random mutations, right? 260,000 plus mutations they were subjected to and were able to find peaks of fitness. And then I showed models in the paper that you cited at the very beginning, in which we had the vast majority of mutations are harmful, tiny sliver are beneficial. And I can show the figures if we want to pull them up. And then you still see adaptive evolution, right? So and I'm arguing that the reason that can happen, right? I'm giving you the mechanism why that can happen is because drift removes most of the neutral mutations, selection removes most of the deleterious mutations, and so what that leaves are the beneficial ones to rise to fixation. Now they do it really slowly. A lot of them are even lost to chance, but that's true of all mutations. And so if the beneficial mutations are super rare, they can still drive adaptive evolution because they can be counteracted or because they're being actually promoted by selection. And that's what our paper showed. And that's what that study with the E. coli fitness landscape showed as well. So it seems like the empirical evidence, as well as the mathematics, the modeling, both seem to suggest that our intuition about deleterious mutations stopping adaptive evolution is wrong. Right? I get why that's an intuition you would have, that, oh, there's so many, it must not be able to work. But if the math shows that that's wrong and nature shows that that's wrong, we should go with those things over what our gut says, right?
Zach, and before you respond, Jerry, because I'm really enjoying this discussion, I want us to continue along these same lines, but just to make sure that the audience and everybody's on the same page on these technical details, it's an important point here. Dr. Hancock, you're saying that the tiny amount of beneficial mutations, in light of all the slightly deleterious mutations, we're still seeing adaptive evolution take place because selection is acting upon those slightly deleterious mutations, while the small amount of beneficial mutations are rising to fixation and novel change. Is that more or less what you're saying, Zach?
Yeah, can I just show this figure, and maybe that'll help if we actually have like a visual here.
Sure.
Can you see this? Do we see this okay? Do I need
Yes.
to make it bigger?
Yeah, maybe zoom in slightly. And then when we're all on the same page, then Dr. Bergman, let's hand it back to you for a thorough response to this. So Zach,
that? Can we see that okay?
That looks good. Yeah.
That looks good? Okay. So I'll just walk through it, because if we're going to talk about it, maybe it'll be useful to sort of explain it for the audience sake. So here in this A panel, this is from our paper. This is the simulation study that we did. And so we picked this distribution of fitness effects from empirical literature. So we went out, we looked at what have labs estimated DFEs to look like? What are mutational effects typically being estimated in like actual laboratory settings? So that's what we ended up using as our guide for our model. So here on the x-axis is the fitness effect. So what is that mutation actually doing? If it's greater than 0, then it's increasing your chance of reproduction. If it's less than 0, it's decreasing your chance. And the percent that it decreases, like 5%, 10%, 5%. Is that right? In both directions. The 0 mark, this is those that are strictly neutral. We didn't simulate any strictly neutral. Every mutation had some effect. 99% of them were harmful. So all the red ones, you can see the large proportion. We actually cut off the axis here. It goes all the way out to like 50%. So there were even some like virtually lethal mutations way out that we didn't show. And then you see this tiny sliver here of beneficial mutations, only 1% of which are beneficial. Now, if you look on the y-axis, what you're seeing are the frequencies of those mutations at the end of the simulation. So everything at frequency of 1 are those that went to fixation that everybody now has, and so the whole population is experiencing. So the red ones are all the harmful mutations that went to fixation. So we see there are some, right? There were some damaging mutations that went to fixation, but notice they're all really, really tiny effects. And then when we go to the other side, like the beneficial side, all the majority of them have been compensated for by beneficial mutations, right? So this side clearly cancels out that other side. And that's why when you look at this population graph here through time, you see growth. And that's the adaptive evolution, that all of these populations were able to grow and adapt, even with this really, really bias towards harmful mutation distribution of fitness effects. So that's kind of just to give the background of what we did.
Okay, so basically, according to what you're saying, most here are slightly deleterious. The small amount that are beneficial, though, Zach, are going to fixation, basically. There's a few, but the effects of those beneficials rising to fixation are providing a compensatory benefit, essentially counterbalancing the potential damage due to the neutrals there in the red. And that results in adaptive evolution where things are still moving forward in a positive manner.
Correct. That's basically it. Yep.
Okay. Well, I appreciate that. That's very helpful to me and hopefully helpful to the audience. Dr. Jerry Bergman, let's now hand it back to you for some thought.
Let's look at deleterious mutations in humans in aging. As a result, you're young, you have very few mutations and somatic mutations. As you get older, you get more and more somatic mutations, and eventually that leads to what they call genetic catastrophe, which is death. And my best example is when my aunt died, went to the hospital and said, "What happened? Why did she die?" He said, "Well, nothing really happened." "Well, what did she die of?" "Well, nothing." "Well, she's dead. She must have died of something." So the doctor said, "Here, sit down. Let me explain what happened. As you get older, you have more and more mutations causing the heart to be less effective, causing your skin to be less effective, causing your brain to be less effective. And after a while, the accumulation of deleterious mutations causes the body to be problematic. And what happens is you have a heart attack, and the heart attack is not what actually killed you. What killed you is the accumulation of mutations throughout the entire body, your brain, the kidneys, etc. And so on the death certificate, we put down myocardial infarction because they know what that means. We're not going to list accumulation of mutations in the kidneys and the spleen and so on. Simply accumulation of mutations, what causes aging, and we know that species age as well as people. And we can see that one reason for extinction of some species is accumulation of deleterious mutations. And so with mammals, with most animals, maybe different E. coli, I don't know. But with what we know about deleterious mutations, it causes a problem, especially near neutral. It adds up, and therefore your entire body is not as effective as it used to be in doing the things necessary to stay alive. And so mammals, multi-cell organisms, the accumulation of deleterious mutations is even mildly and near neutral. Still, it adds up and you end up with a problem. You end up dying. And that's one reason why people only live so long. Well, you see the same thing is true in other organisms as well, especially when it's a mutation in the gametes. Then you have, no problem, but my daughter has that mutation. She has a couple more mutations. Then her children have not only the mutations that I gave her, but also the new mutations. The mutational load increases until you end up with a child that is poor health. And so natural selection, of course, takes care of this. What? Of all 100 conceptions, something like 50, I don't remember the exact number, but something like 50% do not make it to term because the accumulation of mutations produces a level of problems where if the child was born, it wouldn't live long anyways. And so it's the way the body has of natural selection of the fetus, so that if the mutations are at a certain level, the body figures this out and spontaneously aborts the fetus.
Yeah.
Okay,
So
actually, Zach, before you respond, I'm going to give you lots of time to respond, Zach. Just again, to make sure that we're all on the same page, myself and the audience. Would you say, Zach, that the genetic entropy thesis, essentially, that Dr. Bergman just described, is countered largely by this modeling that you've done with Dr. Dan, that you're representing here on the screen in your paper, that basically the nearly neutral mutations do accumulate, a few go to fixation, they're damaging, but the tiny amount of beneficials that go to fixation essentially counterbalances the problem that Jerry is describing? Is that more or less how you'd respond, or what you're thinking, Zach?
So I want to address the idea about aging first, actually, because I actually think it's not the correct analogy, because you don't actually die because you accumulate mutations in life. That no model of senescence assumes that. If that were true, then all organisms would age at the same rate, and yet we don't. Right? You live longer than your dog does, and yet your dog's cells divide at the same rate that yours do. And so the reason that you age is not because you accumulate mutations. The reason that you age is because you have genes that behave differently depending on age and depending on when selection can act on them based on when you can reproduce, like what the age you are at reproduction. So we live longer than our dogs because we don't mature until we're teenagers, right? We don't reach sexual maturity until we're teenagers. And so there's strong selection for any gene that might start breaking stuff, that might make you age. Like if you're a male like me, might make you go bald, right? Like those sorts of aging things. Any gene that infects that early on, selection gets rid of. But the later the action of any gene starts to act and starts to have these sort of harmful effects, selection becomes less able to see it the further you get away from when you started having kids, because you get old enough, you've either died by chance, you've got an infection that's killed you, you've already had all your kids, right? And so the reason that's why we age, not because we're increasing the number of deleterious mutations, but because what's already in us, we are born with a ticking time bomb that selection can't get rid of, because we're in selection shadow when we're older and we've already had our kids. Now, this explains why you age differently than your dog does, right? Because your dog sexually matures much faster. And so any gene that your dog might have that starts impacting it negatively early in life, right, can still survive because your dog is already sexually matured. Right. So I just want to show that the theories about senescence are not typically about increases in mutation. So genetic entropy and aging are two very, very distinct things, and I think we shouldn't conflate them. And so when we're talking about genetic entropy in this context, then yes, what Donny said is correct, that while our intuition seems to lead us to believe that if we're getting all of these deleterious mutations, well, they must just overpower the beneficial ones. But the power of science, the power of mathematics, the power of empirical observation is that we should not trust our gut, right? If science has taught us anything, our gut is not what we should trust. We should trust what we can observe in nature and whether or not what the math is telling us. When we sit down and we model things, what does that show us, and does that match nature better than our gut does? And I think this does, especially when you compare, and this as in the model, especially when you compare this to the E. coli study that I showed, when you compare this to the protein interaction study that I showed, where these proteins could evolve step by step. I think when you take this with the empirical data, I think it's really strong evidence that we have these compensatory mutations that make up for the deleterious mutations that do go to fixation. And then we still have leftover beneficial mutations that are able to drive adaptive evolution if you can adapt further. Eventually you do reach a peak and you can't adapt any further in that environment, and all mutations at that point are deleterious, right? But since the environment is constantly changing, there's always a pressure to keep chasing the next peak. So I hope that makes sense.
Jerry, back to you. Go ahead.
There's another factor, obviously. Rats live what? 3 years. And tortoises live for 450 years. So I know there's a genetic factor that is important as well. But on the other hand, still, if you do any research on aging, you work in a hospital environment, the idea that the gene somatic mutations accumulate is clearly an important reason. And this is why cancer, etc., is often a disease of aging. Not many, or too many, I guess, but not many 5-year-olds die of cancer when they're 5. And as you get older, the likelihood of developing cancer is greater and greater, because indeed cancer is a result of gene mutations, not just one, but several. In fact, most cancers require 3 different mutations to produce the cancer. And so therefore, when you're 25, you may only have 1. When you're 45, you may have 2. When you're 50, 65, you may have 3. And as a result, you develop cancer. So the accumulation of mutations is still a major reason for the deaths of many animals. That's
for
true.
Cancer, but not for aging, right? Those are 2 different things. Like you can get cancer at any age from mutations, but cancer is not synonymous with aging. Aging and senescence is an actual completely different thing. Where like you're breaking down because of the interactions of genes, not because you're like covered in cancer from head to toe.
Yeah, but a factor that predisposes one to cancer is your age. Sure. And that predisposes you to a lot of diseases. I know, leukemia can affect 3 or 4-year-olds. That's true. But on the other hand, that's not normal. That's not common. And that's so the accumulation of mutations is a major reason in the medical field, a major reason for so many diseases that we have. And so many conditions that we see, and you and I, are a result of aging. They're not found in younger people because they haven't accumulated the mutations to cause the problem. I have, for example, predisposed to osteoporosis, and the doctors made that very clear. That's a result of aging, that the accumulation of mutations, your body is not as effective as it was when you were younger in repairing and maintaining bone health, period. And so over and over in medicine, you get mutations are critical in causing so many problems. That's true. You have people who are 95 and can jog around the track and don't have any signs whatsoever of osteoporosis, but we're looking at the majority of people. We're looking at the accumulation of mutations, which is what causes aging. And I think you're denying reality when you don't look at that fact. And those of us who are getting older can see that. And we can see the effect of aging in so many areas. And when I go and visit my doctor, he can say, well, this is a problem as a result of accumulation of mutations. You're getting older, and older people have these problems. Younger people don't because they don't have the mutation. Although, again, got tortoises that live to be 450 years old. You got mice that live to be 3 years old. And so there is a genetic component. But on the other hand, there's still a difference. The average, large dog lives to be 15? Well, no, not 15, I guess, 12. Some dogs, because of accumulation of mutations, die at 12, at 10. Some dogs, we just talking to a friend, dog lived to be 17 years old.
Yeah, I think we've hashed out the effects of mutations for sure, and that clearly mutations have some impact on aging, in the sense that you increase the probability you get cancer as you get older because you're increasing the chance of getting those mutations, right? So we can agree on in that respect. And I think maybe, Donny, we've drilled the genetic entropy as far as we're going to go. So I don't know how much time we have left, but if Jerry wants to lead a question, I'm happy to fill that.
I want to know how you can get a Great Dane and a Chihuahua and how they can interbreed and the dog works. It's like taking a novel, 2 different novels, and blending them together, and you end up with a new novel that makes sense. It's baffling, and I've asked other people, and they have no idea. You as a population geneticist must have some explanation for how you get animals that are so different can breed, and yet the results are pretty good. Best example, of course, is dogs. And I think all the dogs that I've had were a mixture of a poodle and some other animal, and it turned out great. And sometimes I know you have problems like a weak system where they end up with problems with the hips and so on. But by and large, in most cases, and we have we have now over 500 different breeds of dogs interbred. How do you end up with such a healthy dog with combination, and it seems to fit like our Great Dane and Chihuahua mix?
Yeah. I mean, I'll try to take this from a population genetic perspective as best as I can. I love that my degree has reduced me to dog breeding. But I think that the interesting point about dogs specifically is most dog breeds are actually just like incredibly inbred, right? So that's effectively what a dog breed is. This is a highly inbred population. And so by actually mixing them, you tend to get better dogs. I think this is a thing that most people just recognize that have had dogs, right? Is that if you find, a mixed breed dog, they tend to be better, they tend to be healthier. And it's because you're actually reintroducing a lot of variation that can mask some of the things that appear deleterious in the homozygous state that a lot of these sort of like highly inbred or quote unquote pure breed dogs tend to have. Why a Great Dane and a Chihuahua can mate, I have no idea. I don't understand the mechanics of that. Like I don't understand the physics of it, honestly. So it seems like maybe they had help. Like that's the only thing I can think that pulled that off. But really, all dogs are pretty closely related, right? Like dogs evolved from wolves 10,000 years ago, maybe 15,000, like not very long ago did dogs diverge from wolves. So they are still very, very similar to each other.
That's true. But still, you take other animals and you end up with a combination that actually works, even though people think it couldn't work. And you get donkeys that can't reproduce. But on the other hand, you can take two animals that are very different, not just dogs, but other animals that are very different. And you end up with a resultant of a new animal, which has some of the traits of one animal and other traits of the other animal. And how could they blend? Again, taking two different novels, put them together, and the new novel from the two different novels which you put together makes sense.
Yeah, I think plants are maybe the best example of this, right? Like the reason, like, I don't know, I really like flowers. Like the reason like a lot of ornamental flowers and stuff exist is none of them are like natural species. The vast majority of flowers, like roses and stuff like that, are hybrids. They're grafted on from a bunch of different species. That's how you get all the diversity. You're just hybridizing different rose breeds. Yeah, I don't know. I mean, we're to evolve reproductive isolation, to actually be isolated, takes time, and it takes very specific kinds of mutations that have to interact epistatically and be damaging to each other, and that just takes time. And different species evolve at different rates. And most of the ones that we've been tinkering with, we haven't been doing it for very long, right? Like we've domesticated most things fairly recently in agricultural history, right? And they just don't have the kinds of reproductive isolating barriers that you see among other natural populations. So yeah, you're right. It's interesting. You could mix a Great Dane and a Chihuahua, but if you tried to breed like Drosophila pseudo-obscura, which is like a fruit fly, with Drosophila melanogaster, which is another fruit fly, they don't produce viable offspring, right? So the fact that those natural species are incapable of breeding, though they look extremely similar, and yet Chihuahuas and Great Danes, that's a fascinating component of genetics.
What seems to me is that there must be some mechanism which allows basically these genes to mesh, and yet it doesn't cause, in most cases, doesn't cause a problem. It's almost like there is a gene area for color, and therefore you can have several genes go in that area and produce a blend of the colors. So this gene for color goes in that area for color, and therefore you can have a gene for white and black, end up with gray. And so it's almost like
these are alleles, right? These are what alleles are, right? So you've got genes that contribute to color. Most color is actually polygenic, believe it or not. There's many genes that contribute to it. The more genes that you have contributing to it, the more alleles you can have, right? So if you've got 10 genes, two different alleles at each one, you can start getting all kinds of wild combinations of colors, right? And this is true for color. This is true for petal length. It's true for fur length. It's true for every kind of trait you can imagine. You can get lots and lots of variation. And like, quite frankly, like to bring it back to our discussion about evolution, like that's why you can get such immense variety of organisms, right? Is because the underlying genetics enables such immense creativity in the hands of mutation to generate such wildly different varieties that then selection can pick through and say which one of these works best in the environment that we are in, right? We humans, we like to get the pretty colors. We like to breed things for our own purposes, right? But nature, mutation, and natural selection have been doing it for much longer and has produced the variety of organisms we see around us.
To me, it's evidence of design. That animals are designed to blend from two different animals. And you have, again, and I can't remember, you have lions and tigers that can intermix, and you have zebras and I forget donkeys that can intermix. You have a lot of varieties you wouldn't think would be possible, but yet they do fine.
Yeah, but lions and tigers produce a sterile males, right? They follow Haldane's rule. So
Yeah,
only
they can't
only the females
produce.
are fertile.
They can't produce offspring.
Yeah,
Like
the females are fertile, but the males are sterile. Yeah. So I mean, I guess if we still have 5 minutes left, I guess I can ask, what do we mean by the evidence of design? Like what about the ability of two organisms to hybridize allows, implies to you that those organisms were designed? What does that mean exactly?
It means that basically it's designed so that this organism and that organism can mate and produce a viable result, which is what you're looking for. And it doesn't end up with, when two organisms, it's the fact that they can mate, I think, is intriguing. And we've been able to produce so many variations of organisms that we didn't think could ever reproduce together, like Great Danes and Chihuahuas. And the fact that they can do so, I think, is evidence of design. And also,
we designed Great Danes and Chihuahuas, right? Like Great Danes and Chihuahuas aren't things that God designed. They're things that we designed. God designed wolves in your worldview, right? And then we designed Great Danes and Chihuahuas from wolves. Presumably, that's what you would say.
Genetically, they're obviously quite different in so many ways, Great Danes and Chihuahuas. And that is true with so many animals. They're so different, but yet they can mesh and produce results which are favorable for whatever we're trying to do, color, variation, and so on. And there are many. Well, you think the best example you brought up is plants. Plants.
So is it not a more scientific conclusion to draw that we have the actual natural processes that generate this variation that we are then selecting through, right? So when we take organisms and we domesticate them and we breed them, we're actually also weeding them for new mutations as well, right? Like we're constantly looking for new mutations that occur that we can then pick at and drive to fixation, right? Like we are also acting as if natural selection and grabbing those mutations as they occur. So we see that happening. We see mutations occurring in laboratory populations. Why would we think that the natural variation occurring all throughout the rest of the biological world is not the result of natural processes that we have actually observed, and instead assume that it's the result of some process or some entity that we've never observed, right? Like that's the very definition of introducing an unobservable, untestable hypothesis into science by calling down a supernatural entity to explain what can be explained with natural processes.
You call it natural, I call it design. So much of what you call natural, I say are design.
So is a rock designed? Are rocks also designed?
Well, the parts of the rock are designed.
So how do I tell what's designed from what's not designed?
Well, of course, I would say life is designed, and the components of a rock were individually designed. The iron, the minerals,
so atoms are designed. So you think that atoms are the base, the fundamental level of design is the atom?
I would say, yeah, atoms are designed. You're not going to have life on the earth unless you have a certain type and number of atoms.
But we see atoms form naturally, right? Through fusion processes. We know that these can form, in the center of the earth, for example, right? Like in stars when they explode and things like we know atoms are being fused and being formed in these processes naturally, right?
Mm-hmm. Well, you call it natural.
That then implies that we have a natural explanation for how atoms form, and we don't need to invoke a supernatural thing to explain atoms.
Except, why do they form? Why do electrons behave the way they do? Why do neutrons behave the way they do? Why do, I know you talk about quarks and you go into details, but on the other hand, I see the chemical world. I taught chemistry for many years. The chemical world I see is clearly evidence of design, how it works and why it works. And we don't understand a lot of how it works. For example, now we've got string theory and we've got all kinds of new explanations which attempt to explain how they work. But the fact they do work, and the fact is, to me, it clearly appears to be designed, that molecules are designed in essence, in the end, to support life. So I would say the creator said the end goal is humans. And how do we get there? Well, we have to start out with quarks, if you prefer. And we've got the up, the down, the strings, the charm, and so on. We've got quarks. And so that is the beginning of producing the world, which will produce humans. So the goal is humans, and that world has to be produced to produce course, I see the world from a design perspective. Didn't used to, but more and more as I do more and more reading and research, I see clearly the world is designed in so many ways. We say it's natural, but natural doesn't really explain it. I think design explains it much better than to say, well, it's natural. Just the way I see the world, I guess. And as most scientists throughout history have seen the world in that way. It's only until Darwin came along and a few others, Lyell came along, that
it was long before then that people started saying we need to rely on naturalistic processes. Like we can go back to Laplace, right, who also said the same thing. I mean, and Newton also only used natural processes in his scientific work as well. So like I think we don't have to hash out the history of the development of science to agree to disagree. That I don't think that we can actually. I don't think you're capable of telling me how to differentiate between what is designed and what is not designed when we're looking at the natural world. But maybe that's like inferring design could be a whole topic for maybe another day.
Laplace was a renegade, but on the other hand, I think Newton clearly saw the world as design, as so many other, Aristotle and so on, saw the world as design, and they saw the world as being produced for a certain purpose, a long-term purpose. And yeah, I know Laplace is a scoundrel, but he's one of the first. There's a few others. But
Laplace was a scoundrel. That's why we like him.
Gentlemen, that was an excellent discussion. We even went over by a few minutes. I think we were comprehensive. We touched on many important topics and issues and went back and forth on some of the most important issues as well. We have closing statements and then we have Q&A. To make this discussion as complete as possible, I had one question to throw in there, and it was based on something that you argued closer to the beginning, Dr. Hancock, when you gentlemen were discussing the nature of gene duplications. I had a clarifying question. Zach, you pointed to, and correct me if I'm wrong on any of this, you pointed to fruit flies, where we can know that gene duplications occurred. We can infer this basically by using genome sequencing technology, where we compare the genomes of these different fruit flies. You pick fruit flies largely because as creationists, we may agree that these fruit flies are related. So we can compare these genomes and compare these species and individuals and see a region that's maybe in one individual that's not in another, with a novel function, basically inferring that this was duplicated, followed by neo-functionalization. Is that more or less what you were saying, Zach?
So the way it works is that you've got the two species, and then we know that they had a common ancestor, or I assume we're all agreeing that they had a common ancestor. And so you're looking at that specific gene that they both share, and we call that the parent copy. And then in one of the lineages, either in Melanogaster or in Pseudoobscura, along that lineage, a duplication has happened. And we know it happened along that lineage because it's young. And what by young, we mean that it's a lot more related to this copy than it is to the homologous copy in the other species. Does that make sense? And so we know that it's a young duplicate. And then we basically say, okay, in this daughter copy, does it do something different in this fly than the parent copy does in the two species, right? So we know what the parents' copies do. Does the daughter copy do something different? And they found 281 examples in which the daughter copy did something different.
Okay, so in those 281 examples where the daughter copy to the parent copy did something different, had a different function, then your inference is, and I'm not saying it's bad or good, that's why I want to hand it to Jerry to interact with just for a second here. Those 281 instances would be novel functions. They evolved through duplication followed by neo-functionalization, essentially.
Correct. Yeah.
Okay. It's a fair point. And so I'm curious, Jerry, as a creationist or arguing for your position tonight, I'm curious as to your overall thoughts on that and what would be a good counter if you disagreed with the inference.
Well, there certainly is a lot of flexibility in organisms, especially fruit flies, which is why they were so useful in studying mutations and learning about mutations. And a lot of organisms have a lot of traits which are very useful to do research. And so I don't see that as any concern, I guess. The fact that duplication can occur, and I have no doubt that it can occur, but in most cases, it's not. It is a problem, or at least in our large number of cases, it doesn't solve a lot of the problems that they have in talking about the origin of species. And of course, Darwin's whole book was titled The Origin of Species, but he never really talked about that. But Darwin was mystified. He recognized that he had no explanation for the origin of species. He had no explanation for the origin of new genetic information. He just had no idea. He just thought there was naturally enough variation built in that we could get evolution from the natural variation which is already there. And so the problem we've had, which is what this debate focuses on, is the origin of new genetic information. Where does it come from? And my conclusion is the only explanation is it was created. That natural processes do not explain where it ultimately came from. And Darwin turned out to be wrong, and there's a limit in variation. Breeding horses, we can see. You can breed until you get so fast, but to breed just slightly faster has been found to be difficult. You can only go so far with breeding animals. And once you reach that limit, that's it. You can't go farther. Of course, Darwin felt there was no limit. You can just keep going beyond that
Okay,
limit.
I'll jump in real quick. I appreciate those points. One final question, and then Zach, we're going to throw it to you just for a 20-second final word to be fair, and then we're going to jump into closing statements. And so, Jerry, in regards to the fruit fly example, gene duplication followed by neofunctionalization, are you essentially saying that perhaps in some cases gene duplication could result in novel functions, but that ability is built in somehow, that these traits are built in to be manifested through this genetic process?
Oh yeah, I have no problem with that. I think that's probably the system is built in so that these changes can occur and produce genetic variety. But again, there's a limit of how much genetic variety you can get. So yeah, I have no. Once it's proved in the lab, I'm okay. Once it's proved in.
Okay, that helps.
It was proved in a lab. I gave you laboratory examples that it was proved in a lab. And it's also curious that it's built in to generate variation. So it's built in that mutations happen, or that mutations are capable of happening, but also mutations are bad. So do you see how that's kind of a conflicting statement, to see that like God developed it to be able to generate variation? That is what a mutation is. But then all mutations are bad? I don't understand that logic. To me, if we see mutations occurring, we see them in the lab, we see duplications occur, right? Then it's the natural process that is producing that variation. There's actually no need to invoke anything supernatural to explain what we see happening just within the lab itself.
Yeah, I'm not saying they're always bad. I'm saying some mistakes can occur and the results can be good. So, but as a whole, mutations are problematic, and we need to avoid them. If they were beneficial, we would recommend young girls who haven't had a child yet, young men who haven't fathered a child yet, go to your local hospital and sit down on the CT machine and spend 20 minutes getting some good amount of radiation so that you can increase the level of mutations, and maybe you'll have a genius. What you'll have is, though, if you went to a hospital and talked about this, they probably would have the guys in the white coats come in and take you away and wonder what's wrong with you.
Now you know that's preposterous. You know, I've already agreed that the vast majority of mutations are deleterious, and I would never say people should go sit in front of an X-ray machine. Even Herman Mueller knew that X-rays were bad. So that's a straw man.
Gentlemen, we could probably continue this for another little while, but let's wrap it up there. Again, excellent discussion. I do appreciate how professional the both of you were tonight, especially during the discussion, and just how much we covered. I really enjoyed this, and it also helped me greatly to understand a lot of the points that have been made and that have been made outside of this debate as well, including your paper there, Zach. So, okay, gentlemen, closing statements. In light of everything discussed and interacted with tonight, great opportunity to essentially land the plane on our respective positions tonight, address anything we feel has been left hanging, and leave no stone unturned tonight on the issue. So, Zach, we are going to hand it to you. You get the first 5 minutes for a closing statement, and I'll start the timer on your first word. Go ahead.
Okay, so again, thanks everyone for being here. Thanks, Dr. Bergman. Thanks to Donny for setting this up. This was a lot of fun. What I really, really hope that you as the audience took away from this, so the whole reason I'm here is for you, the audience, right? Because what I do, the reason I do this, the reason I have a YouTube channel is because I care about education. Like fundamentally, that's the thing that interests me the most, is for you to come away and learn something about biology, right? And so I hope that what I have shown you today is that evolution is a scientific principle, is absolutely capable of explaining where new stuff comes from. Right? It's capable of doing this using natural processes that don't rely on mysticism or storytelling or any fables or anything like that. It's relied on things that can be tested, things that can be observed within laboratories and in natural populations, and are capable of driving the variation that we see in populations. And I've given you several examples of that today. I've given you how new genes can emerge in step-by-step Darwinian fashion. Right? You can go from two proteins that must interact with each other all the way to a completely different set of two proteins that have to interact with each other, one mutation at a time, never disrupting function, right? Like that's a pretty powerful mutational demonstration that you can get new stuff emerge without ever breaking anything. Right? I've showed you that thousands of brand new genes can emerge, including thousands of brand new regulatory genes can emerge via the neo-functionalization process, where you have an original gene, that gene duplicates, and then that duplicate copy takes on a novel function. Right? So all of these things we have been observed. I gave you empirical studies of this, and I also talked you through some theoretical results where we showed that even if the vast majority of mutations are deleterious, a tiny sliver of beneficial ones can not only compensate, but can actually drive adaptive evolution. I showed this mathematically using the modeling approach that I showed on the figure before from our paper. And then I also showed this empirically with the E. coli example, where it was really, really easy. There was over 100,000 mutations that would bring you to that peak of fitness. So mutations are absolutely capable of explaining Hugo de Vries's cutesy phrase, the arrival of the fittest. Thank you so much all for your time. Thanks again to Dr. Bergman and to Donny, and I will see the rest of my time.
Dr. Zach Hancock, thank you very much for that 5-minute closing statement. This means to the audience, we have 5 more minutes for you to send in your last-minute questions, challenges. Perhaps you've heard something tonight that was said by Dr. Hancock or Dr. Bergman, and you disagree with it and you want to challenge it. Feel free to send those challenges in the form of a question, of course, as well as objections. And we are going to interact with your comments very soon in about 5 minutes in the Q&A. Dr. Jerry Bergman, we are now going to reset the timer and hand it back to you. You have 5 minutes. Go ahead.
Yeah, I would say very clearly from the work that I've done in medical school and in the area of mutations that as a whole, they're deleterious. You do not produce people by breaking things, by causing damage. And the damage to mutations, which result in mutations, is basically you're breaking genes. You're damaging the gene as a whole. I'm not saying a few rare effects. Sometimes you get some beneficial results. Sometimes it helps the organism in some ways. But the vast majority of time, what I'm saying is mutations are a problem, causing disease, causing mutations and genetic mutations in the gametes, and as a result, you have children that are born, babies that are born with various diseases, etc. And so mutations are a major cause of disease, a major cause of most diseases. As I mentioned, the major cause of diseases are toxins and mutagens, and by far the most important is mutations. And so by far the most important cause of disease is mutations. We know that. That's commonly understood in medicine. And the area I worked at in the medical school was in the area of mutations. And it was very clear from the work that we did that we're talking about harming, not helping, but harming the organism. And therefore, mutational load must be kept as low as possible. You must take care of yourself. And that's why hospitals have certain requirements. When they do an x-ray on your teeth, they cover your body up with a lead shield. That's why the x-ray tech leaves the room, turns the machine on, does the x-rays, to avoid the likelihood of her being exposed to certain level of mutations. That's why they wear a badge, which measures the alpha, beta, and gamma level of radiation, and it must be kept below a certain level. If it doesn't, they take you out of that area and put you in some other area of the hospital. But of course, the goal is to try to keep it to a low level, ideally 0 level, but that's not possible. So therefore, as low as possible below certain standards. And so it's clear from the medical field that mutations are a problem, a major problem in causing disease. One of the major causes of both hereditary and somatic diseases like cancer is mutations. And there are about 5,000 different diseases now that we know that are the result of mutations. So as a whole, it is harmful. And yeah, you can pull out a few positive effects with E. coli or other microorganisms or fruit flies, etc. But by and large, mutations are a problem that you must work hard to keep as low as possible. And that's why we were so careful in working with radioactive substances like iodine-131 and so on. So we were less likely to be exposed to a level which would cause mutations. Mutations are not good. A mutant is not beneficial. And to conclude that evolution as a result of billions of mistakes is a major problem. And one tries to find examples, try to work around this fact, but in time, I think it'll become more and more obvious, and evolutionists will, like they've done with the progression. They realize the progression is not true, it's false, and there are problems with the data behind it. And I think eventually we will understand that, yeah, you get a few exceptions, but by and large, the fact is that mutations are not a friend of life, and therefore a problem, as is well recognized now, at least in the medical field. And therefore, I enjoyed, by the way, with Zach. He's a smart guy, does a good job. Of course, he went to U of M or did work at U of M, which I'm very familiar with. I did work also at U of M, so I'm very aware of the school and good people they produce. Not all, but generally good people. So therefore, I appreciate being able to interact with him. And I have to admit, I've learned some things from his presentation, and I enjoyed working with him. By the way, no name calling, which was nice. Another person basically said, yeah, Bergman has 9 degrees, but 1 is from a diploma mill, which bringing those things up or making those claims, which are false anyways, but it causes me to lose respect for the person. We need to deal with the data, with the facts, and Zach did that. And therefore, he is to be commended for sticking to the topic and a debate where both he and I can learn and grow from, which is one of the purposes of this whole format. And I appreciate that a lot. And I'm very thankful to him. Nice guy. He's someone I think you could go out to a restaurant and have dinner with and have a good conversation. And so I appreciate that.
I told you he was a nice guy, Dr. Bergman.
Yeah, you did.
You didn't believe me. No, I'm just kidding.
Yeah.
My reputation precedes me.
Yes.
Yes, I was telling Zach, he's one of my favorite evolutionists, telling him backstage. So, gentlemen, this was great. A name-calling free debate, right, Dr. Bergman? So this was good. And it's one of the reasons, Jerry, what you said, that I love to host so many debates every single week, several debates, because it's a great learning opportunity. I always learn a ton, and I learned a ton here. So Zach, Jerry, fantastic job. Great debate. To the audience, share this around, and let's keep the discussion going. Okay, gentlemen, that was 5 minutes each, and we are now moving into some questions, some Q&A. How we will run this Q&A is whoever the question's for gets the first word, of course. Let's say it's for you, Zach. You get to respond. Then Jerry can provide his thoughts, a rebuttal perhaps if he'd like to, and then we throw it back to you, Zach, for the final word. So essentially, whoever the question's for gets the final word. And okay, let's go through our questions here. And we can start with Andrew Cumming. We've had a good mix of both sides represented in our live chat tonight. Andrew Cumming has a question for Zach. How well do we currently understand the evolution and genetic pathways behind brains in different animals? And what avenues of further research are needed?
Yeah, so that's a very interesting question. So brains are so maybe we explain what a brain is. So brain is essentially a highly concentrated vat of neurons, of cells, of very specialized cells, right? And in different kinds of organisms, the brain is less well developed, as you would imagine. And then in many organisms, there are no brains at all. It's just like a glorified bulb. Especially in like flatworms, for example. We've talked about flatworms a lot here. Love it. Flatworms is effectively just a little bulb. In other organisms, it's just kind of a ladder-like structure. So what counts as the thinking part of the organism, it's really the part of the organism that can respond to the environment, is highly variable. In terms of our understanding of its genetic pathways, there are people, I'm sure, that do know about the genetic pathways of the development of the brain. I am not one of those people. So I would recommend, if people are interested in that, to go read like evolutionary developmental like Evo Devo stuff, developmental biology stuff, cell biology stuff. Go check out those sorts of journals. It's a little outside of my expertise, so I'm gonna sort of bunt on that.
Thank you very much, Zach. Jerry, over to you.
Yeah, appreciate his comment. So don't really have much to add, except the brain is over and over said to be the most complex structure in the universe, enormously complex. And it's so complex, we have yet to fully understand it. We need to understand it more because we have certain diseases which are a problem, like the disease of the brain of aging, caused by accumulation of mutations, by the way, and don't understand what caused these mutations and why they cause a problem. Like Huntington's chorea, of course, which we do now understand more. But the Parkinson's disease is a major concern, and Alzheimer's, I know that because my mother had both Parkinson's and Alzheimer's disease. And therefore, I've tried to follow the research in that area. And basically, we have some good ideas and still have a lot to learn about what causes these. But the excuse is given is basically it's the most complex structure in the universe. How do you expect us to understand this within the next 30 or 40 years, which we've been doing research on the brain? It may take another 40 years to fully understand how the brain works because it is, well, the most complex system in the universe. And one might wonder, how in the world could the most complex system in the universe evolve through accumulation of mutations? When mutations cause Alzheimer's and Parkinson's disease. And that's my thoughts.
Thank you very much, Jerry, for your thoughts. Zach, you get the last word if you'd like it. Question was for you.
I guess I'll just give a pitch for scientific research. We need a lot more of it. As much scientific research as we can. Fun science. That's my pitch.
Thank you, Zach. Another question for you, Zach, from Prairie Christian Outreach. Can we scientifically show that the information in DNA and epigenetics can be the result of a random process?
That was for me, did you say? Yeah.
Yes, yeah.
Yeah, so great question. I'm going to try to not get hung up on the word information here and just assume that you mean DNA that can specify new things, can specify a new protein, right? Presumably that would be new information if DNA can specify a new protein, right? And I've given you several examples of exactly that, right? So you effectively take mutations we know are random. There's been a gazillion studies going back to the 1940s and 50s that demonstratively show that mutations are random with respect to their impact. Not with respect of where they occur, but with respect of the impact that they have, they are random. And so in effect, what I've shown in studies just tonight is that you can take a gene, you can duplicate it, which is a mutation, and then random mutations occurring to that gene can generate novel functions, right? And I think even Jerry has agreed that that can happen sometimes. And so it seems like you can, with random processes, increase the information in DNA. Scientifically show it, I think that's exactly what I demonstrated in the studies tonight.
Thank you very much, Zach. Jerry, anything you'd like to add to that one?
Yeah, we know I like to use the word information because indeed DNA is information. It's a code and it has an enormous amount of information. And the source of information we know is intelligence. There is no way that information can be produced by random processes. It's a result of intelligence. And even evolutionists recognize the fact that there's a huge amount of information in DNA, and this information indeed is the result of, they wouldn't go this far, but result of intelligence. And indeed, people who do research, and by the way, I was involved in DNA sequencing and trying to understand specifically certain genes, which when we work at the medical school, and clear to us and the people in the lab that we are looking at information and what it can do and what it can't do. And so I agree that this is information in DNA and epigenetics. I didn't raise that, and Jack didn't either, but on the other hand, epigenetics is interesting modifier of this information, or turning on and off parts of this information. We have a ways to go before we fully understand the information in DNA in the brain, especially.
Thank you, Dr. Bergman. Dr. Hancock, you get the last word.
So I thought Jerry and I agreed that you can get new proteins via mutation. Maybe we don't agree, or maybe we're disagreeing on what information means. And this is maybe the problem. Information is a squishy word that can mean many different things, and it seems to mean whatever you want it to mean at any given point. All I care about as a biologist is, can you generate new proteins? And mutations can do that. So
thank you very much, gentlemen. Along these same lines, on the same topic, we have a question here from Andrew Cumming, but this time for Jerry. And so the chat has been discussing beneficial mutations throughout the debate, of course, no surprise, largely the topic of tonight. And Andrew is wondering about the many occurrences of anti-freeze proteins, these novel proteins, it's argued, Dr. Bergman, in ice fish. For example, at the top of my head, I think there's notothenioid, anti-freeze Arctic fish, and cod, perhaps. Jerry, what are your thoughts on that? Is that an example of novel proteins?
It could be, and it's been 5 or 6 years since I looked at that, but it's a minor change that allowed the fish to be able to develop a type of antifreeze, which the molecule, the protein, does not freeze until it's below a certain point. So yeah, that certainly, it could be an advantage. And again, I don't remember the reading I've done on that. It's been quite a while, but indeed that could be an example where indeed a random process changed the protein in such a way where indeed it did function as antifreeze proteins in this case. And I should, I guess, get my notes out and look at it, but that could be a good example. And I'm not saying that there are no examples. I'm saying that there are very, very few examples. In fact, when I looked at the literature, there was about a dozen, which you see over and over and over. And I wonder, is this all there is? I'm sure there's more than a dozen, but these are mentioned over and over. And how many more there are, I don't know. But my guess is we're not talking about enough of these examples to produce, as they say, from a goo to you by way of the zoo. So that's the problem.
Would that be considered new information, Jerry?
No, I don't think it's new information. It's just a change in the protein so that the protein had reached lower temperatures before it froze. So it's a matter of at what temperature it freezes, or the fluids in the fish freeze. And in this case, it can reach lower temperatures and not freeze, not cause a problem. Of course, on the other hand, without this protein in frigid waters, you wouldn't have too many fish leaving offspring. So there'd be a strong selective pressure toward any changes that could allow the fish to live in this ice cold water without producing freezing. But that's a common example I brought up, and I should recall more details. But
I appreciate it, Jerry. Zach, there's some points for you to interact with. Go ahead.
Yeah, I mean, I think this is an example of why the term information can mean whatever you want it to mean, whenever you want it to mean something. I think that like in biology, what we care about is, do new functions emerge, right? And can mutations cause new functions to occur? And like molecules that act as antifreeze in the blood of many fish and insects, like there's lots of antifreeze proteins out there, that can occur is a novel function, and it allows them to adapt and survive in the environment that they're in. right? So that, to me, that is literally what we mean when we say the arrival of the fittest, right? So that is the new information as far as like it can be meaningful in biology. And as far as like, is there enough of these examples? I gave literally thousands of examples in just one study of novel regulatory genes acting and several hundred of novel proteins acting. So
I appreciate it, Zach. Jerry, this one was for you, so you get the last word this time.
Yeah, the information, of course, would be there to produce the protein, but of course the protein has a function which is not related to information, except the information is necessary, and the change of information is necessary to produce the protein, which in this case happens to be beneficial. And you could see in that environment, anything that would allow the fish to live in colder waters would be, or most anything would be advantageous for the fish.
Thank you, Jerry. For those wondering in the live chat, Jerry, if you could answer this, I think, important follow-up question. What would be an example of new information? If a proponent of evolution were to say, what could I show you that would be an example of new information if these novel proteins are not? How would you respond?
Well, information would be the code is producing something that's beneficial. So we would call that information. If it's not producing something beneficial, we would call it gobbledygook. It's not information. It's simply random DNA base pairs or DNA. And so it's gobbledygook, and therefore it would not be information. So I guess if it works, if it produces a protein that's functional, we would say that that's information. But again, in studying DNA, a lot of people use that term because that's really what it is information. I know it implies intelligence behind it, and people don't like that inference, but on the other hand, it is clearly information.
So if a protein involves the ability that it didn't have originally to act as an antifreeze, that's a new function, and by the definition you just gave, is new information.
Yeah, you could call it claim as information.
There we go. We have evolved a new information.
Yeah, in that one case.
So would it be accurate, Jerry, for Creatius to say that there's no examples of new information, or should Creatius say there might only be a few?
Yeah, there's a few. You have to be careful in biology. I'm sure Zach knows. There's always a few examples of just about everything. When you assume certain things are true, like certain organisms can actually live inside of ice and survive quite well in ice. How in the world could that be true? Well, it is. We know there are organisms that live in ice and do well in ice. There's a large number of these. And so almost anything you can imagine in biology, you find somewhere is often or can be true, like ice organism.
Okay, gentlemen, I appreciate you both answering my clarifying questions and helping this to be that much more comprehensive, I think, which is what we want for the audience. Next question is for Dr. Zach. Comes in from Jacob. What do you think about detecting design through detecting a very improbable combination or range and solely it having a function or matches an independent pattern?
Yeah, great question. So there's two parts to that question, and I'm going to answer them separately. So the first part is about, can we detect it, about the very improbable combination or range. The problem with inferring design by things that are improbable is you would infer design from random combinations of numbers, right? If I said, give me a random sample of 100 or 1,000 or any number of numbers, you could just give that number. But the chance that you gave that random combination of numbers is astronomically infinite, right? So it's very, very unlikely that you give me any number or any series of numbers, and yet that's what you gave me. So that's an incredibly improbable event, and yet we do it all the time, right? That just randomly sampling numbers happens all the time. So I would caution against thinking about improbable events as those that necessarily imply design. The second thing is about functions that solely have functions in that one case. I don't know of any example in biology that's like that. And I tried to be sure that I showed you some examples where you could arrive at a fitness peak by many combinations, right? So there were many, many ways to make the same protein and do it well. And in fact, the fact that there's like millions of species on this planet, all of which have our unique ways of making our living and have unique proteins and stuff like that, demonstrates just how versatile biochemistry is, right? Like the fact that we can all do things in so many different ways and still persist implies that there is not just one way to make a living. There's not just one way to make a protein. So, yeah.
Thank you very much, Zach. Jerry, the floor is yours. Go ahead.
Yeah, I think Jack made some good points, so I really nothing to add.
I think you kind of did just answer this, but it is a follow-up from Andrew for you, Jerry. And he's asking, why don't beneficial functional mutations, based on the definition you gave earlier, count as new information then, Dr. Bergman?
Well, I suppose you could have individual cases where there seems to be new information, but again, there's so few mutations that are beneficial. So we're talking about a handful. And when I read the literature, it's always the same, tends to be the same ones. So, but generally, when we look at information, we're looking at something that produces, in this case, a protein which works, which has a function. So, yeah, you could call it new information, and you could give it that term, but proteins we have in our body, there's so many, clearly as a result of clear information which produces a human being, that other codes or damage to the codes would not produce the same human being in this case.
Zach, anything you want to add there before we move on?
No, I mean, I think that as I showed before, I demonstrated thousands of brand new protein coding genes and thousands of new regulatory genes. So there's at least thousands in just a single study of examples of new information. So I see no evidence that it's as rare as we might think it is.
Jerry, would you like to respond to?
Yeah, I did a survey of the literature on beneficial mutations. I looked at that term, beneficial mutations, and did a survey of the literature, and I found there was about, I can't remember the number, but probably 400 and some articles that talked about beneficial mutations. And so most of these that were supposedly beneficial mutations were actually not beneficial, but they were beneficial only in a certain narrow environment. And many of them were beneficial not for the organism. In fact, most were beneficial not for the organism, but for us. For example, seedless fruit that's classified as a beneficial mutation, but it's not beneficial for the fruit, obviously, at all, but it's beneficial for us. And so in doing research on beneficial mutations, I didn't find much evidence at all that there were a large number that Zach implied are there. So at least, now that's a keyword, of course, I used. And so if you didn't use that keyword in the article, it wouldn't come up. But on the other hand, I think there was about 400 articles that did come up. And I did research on most of these and found that the beneficial mutations were not beneficial in the long run and were not beneficial for the organism, but were beneficial for humans. And there are quite a few in that category.
Thank you, Jerry. Thank you, Zach. Next question is for you, Zach. Redefine Living has a question here. He says, evolution is predicated on a working model of abiogenesis. Therefore, evolution cannot explain the arrival of anything. How do you respond to this?
Evolution is not predicated on a working model of abiogenesis. The first statement is false. If you tell me God created the very first cell, God made the very first cell, but so from that, all of the work that I would do in evolution would still be exactly the same. That very first cell is just the progenitor of all living things on this planet. Evolution in no way is predicated on a model of abiogenesis. Now, we would like to know how abiogenesis occurred. We try to study it and understand it, but that the evolutionary process that I laid out, like the theoretical structure that I literally began my presentation with, didn't have abiogenesis in it, right? Because it's not actually predicated on abiogenesis. So the premise of that question is incorrect.
Thank you, Dr. Hancock. Dr. Bergman, anything you'd like to add?
In teaching chemistry, of course, we get into the area of abiogenesis, and it's an area that evolutionists have proposed many theories and attempted to explain possible scenario for abiogenesis. And from my research, it's pretty clear that they have no really clearly viable idea of abiogenesis. And of course, one way of trying to prove it is trying to find life on other planets. or other moons in our own solar system. And so far, although we're optimistic about finding life on Mars, certain other places, we found no evidence of life anywhere. And one reason for looking for life somewhere else is that would prove abiogenesis. And so far, although we have a number of moons of planets that we're looking at, or satellites actually, they're not moons, but satellites that we're looking at, and other planets like Saturn has several, Jupiter has several, that are, according to evolutionists, promising. But my guess is they're not going to find evidence of life on any of those locations because abiogenesis is simply not viable. You have to have a deliberate creator attempt to produce the first living organism. And the first living organism is not, quote, simple, as they often say. There's no simple organism. If an organism is alive, if it can reproduce, it is not simple, period.
Thank you very much, Dr. Jerry Bergman. Dr. Zach Hancock, go ahead. You get the last response.
Yeah, I mean, again, I think that evolution is not predicated on a working model of abiogenesis. I think that there actually are many working models of abiogenesis. I mean, they're just working models. They're hypotheses. And organic chemists can talk to you more about it. I'm a biologist, so abiogenesis is quite far removed from anything that I deal with. But yeah.
Gentlemen, I appreciate the time in light of how busy you both are that you've offered for tonight. It's been a great debate. A few hours has flown by. It's definitely the kind of debate that I will be rewatching a couple times because it was packed with information, and I like the free-flowing nature of the discussion as well. It allowed us the opportunity to cover a lot of important topics. And so, gentlemen, let's wrap up the Q&A and the debate there. Zach, Jerry, again, thank you so much. Let's have some quick final words, final thoughts. Dr. Zach Hancock, your first formal debate officially on the Standing for Truth debate platform. I hope you had a good time. I hope you had fun. Let's hand it to you for some final words, final thoughts.
Yeah, so I mean, I guess it's my first formal debate since high school Lincoln Douglas debate. You know, so it's like the first formal debate in a very long time. No, I had a great time. It was a great conversation. I enjoyed interacting with both you and Jerry. Yeah, had fun. I hope the audience learned something and could come away from this knowing a little bit more about biology and science.
Thank you, Zach. It's important that we, on whatever side, get out of our scientific echo chambers, come together, discuss these issues, largely for the audience. We hope we get people thinking about these issues more and continue the origins discussion. So, Zach, thank you very much for the final words. For those that want to see more from Zach, do check the description box of this video for a link to his YouTube channel. Dr. Jerry Bergman, thank you as well for your time. I really appreciate it. I hope you had a good time tonight. I know I did. Dr. Bergman, let's hand it to you for some final words and final thoughts.
My question is, if we had a recording of this, would it be appropriate to show this in a public high school or college class on biology? And I've asked this to other debaters, and they said no, because we can only, in the high school, college biology class, we can only present one side. We cannot present the other side. And of course, I've presented to a certain degree the other side. And they're basically, the courts are very clear. This would not be appropriate for a college or high school class because, indeed, the other side is presented, and that is not allowed because of separation of church and state, they claim. And so, therefore, we only get one side in the college classroom, and I think that's tragic. I think we should get both sides clearly presented, and that's, of course, not what happens. I have to ask Zach, have you ever heard the other side in any of your courses presented? Have you ever had a professor that came in and, well, let me give the intelligent design side. Let me explain that. Have you ever had that happen?
This has been so cordial. I'm so sorry, but there's only one scientific side. There's a reason that we only teach one side is because only one side is science. So that's why you don't see the other side. The other, like intelligent design, creationism, those are religious perspectives and they have no place in the science classroom. You can teach it in philosophy. I would show this debate in a philosophy class or in like a religious studies class, those sorts of things, but not in a science class.
What about presenting this book in a class? Could you summarize this book and present the information this book brings out? Would that be appropriate in a?
I don't know what that book is, or the contents of that book, or who wrote that book.
Published by Yale University Press. They're usually think they're a pretty good press. The professor is from Great Britain, so that may be a point against him. But on the other hand, I thought he did a good job.
I'll tell you what. Just send me the link to the book, and I'll take a look at it.
Well, just Monkey to Man. Look on Amazon. Gowan Dawson, Dawson, and look on Amazon or look on a number of websites, and you'll find this. And by the way, many other books. I have a whole bookcase full of books which are critical of evolution, written by evolutionists or those who are on the fence. And from your statement you said earlier, would it be appropriate to present this information? Well, you would say probably it's not, because it does indeed.
And that could be a whole nother debate topic.
Okay.
All right, guys.
Hold on. Maybe Zach, or maybe Donnie, you could put information about this book on your website, and then people who wanted to could obtain a copy and read it.
Okay. Ending the debate with a bang. I like that, gentlemen. I enjoy watching you guys interact. I enjoy the banter. You're both good sports, and this was a solid professional debate tonight. I enjoyed it. I hope you in the audience enjoyed it as well. Again, if you want to see more from our speakers, our debaters tonight, do please check the description box for links to their YouTube channels, their websites, so on and so forth. And so with that, thanks again, Zach and Jerry, for a debate to remember on the question, can evolution explain the arrival of the fittest? Standing for Truth is signing off.